well-organized system where we had right from the presentations we're going to happen today to our posters which are displayed outside, and they are of that caliber which really gives us a thought that people are working, and that's the future is going to be totally different. I'm honored to have our faculty from AIIMS, our distinguished guests come today. For every session, we'll have a different group of people sitting in this audience, their discussion, their participation, and we'll are very, very sure that you're going to learn something out of every presentation. And again, on behalf of entire research section and on behalf of Director AIIMS, I'd like to welcome all of you today for this particular function. I'd like to thank all those people who have been behind this success of this Research Day organization. Thank you and welcome all of you.
Thank you, sir. And to set the ball rolling with the first session, I invite Dr. Sanjukta to take over from now. Thank you.
Good morning, everyone. Welcome to the first session of the Research Day, Ignited Minds Best Papers. We'll first have the platform presentation now by PhD students. For this session, it is our honor and privilege to have Professor Y.K. Gupta and Professor V.L. Kumar as the chairpersons for this session. Professor Y.K. Gupta is currently the president of AIIMS Jammu. He formerly held the positions of President AIIMS Bhopal, Dean Academics, and Head of Department of Pharmacology at AIIMS New Delhi, as well as Director of the Indian Institute of Toxicological Research at Lucknow. He is currently the chair of several high-powered committees of the Ministry of Health and Family Welfare, as well as the National Scientific Coordinator of the Pharmacovigilance Program of India. He has also received numerous awards and honors, including the prestigious Shakuntala Amirchand Prize of ICMR. Welcome, sir. Professor V.L. Kumar is currently the Dean of Examinations, as well as Professor and Head of the Department of Pharmacology at AIIMS New Delhi. She has received several awards and honors, including the C.L. Malhotra Research Prize from the Association of Physiologists and Pharmacologists of India. Welcome, ma'am. I now request the chairpersons to please conduct the session.
To be here and in the, hello, is it okay? Good morning, everyone. It is a pleasure to participate in the Research Day activity. Today's first session starts with the presentations by the PhD students. Here we have 3 students presenting the work. The session is being co-chaired by Professor Y.K. Gupta, who's here.
Thank you very much, and it is my privilege to be here to chair this session, and I understand that this is one of the most competitive session. And I believe that PhD students are the backbone of research in any lab. Because when they conduct research, it flows. One PhD students give the bat to the next PhD student, and so the lab continues. PhD students are also the workforce which establishes lab. They establish lab, and I think if there are no PhD students, I would say that the research would be much, much less. And these are the PhD students which are presenting here, so I am really privileged. So I would say that before these three come, I would like to give claps to all PhD students of All India Institute of Medical Sciences. And not only of this institute, but all in any institute, when we say we have these many PhD students, that indicates the academic and research strength. So I congratulate all the three who are here, but I also congratulate all the PhD students of all the departments at All India Institute of Medical Sciences who set up very high standards of academic research. So the first presentation, and I understand that the students, the PhD students are aware of the rules, and I see that only 7 minutes are allowed, and there are 2 minutes for question-answer session, and there should be some timekeeper. So I would suggest that after 6 minutes or 6.5 minutes, you give a warning signal, maybe by placard or maybe by a whatever method you have devised. So the first presentation is by Department of Pulmonary Medicine by Jaya, and the title is Clinical Application of miRNA in Exhaled Breath Condensate of Pulmonary Tuberculosis Patient, a pilot study. Over to Jaya.
Good morning, all of you. First of all, I would like to thank all the committee members for selecting this abstract for the platform presentation. I am Jaya, PhD student from Pulmonary Critical Care and Sleep Medicine Department under the supervision of Professor Anant Mohan. Today I am here to represent this study, Clinical Application of microRNA in Exhaled Breath Condensate of Pulmonary TB Patients, a pilot study. Pulmonary TB is a major public health challenge globally and is the leading cause of death with a single infectious agent, Mycobacterium tuberculosis. Still, TB remains the top infectious disease. According to global data, around 1.3 million people were died due to tuberculosis in 2020-22. The reason behind this, 40% of the TB cases remains undiagnosed, and among multi-drug resistant TB, this percentage jumps to 80%. Around 1/3 population are infected with latent TB. However, only 5 to 10% people have active tuberculosis in their lifetime. The basic diagnosis for the tuberculosis is radiological investigations, chest X-ray, CT scans, molecular investigations like gene expert, PCR, microbiological investigations, Jaden stain, MESIT, LJ culture, etc. The sample for the pulmonary tuberculosis is bronchoalveolar lavage, expirated sputums, induced sputums, gastric aspiration, and nasopharyngeal aspiration, etc. Some of these samples are very invasive and needs a super specialty setup and expertise to do. So therefore, we need a non-invasive sample for the detection of tuberculosis. So in this context, exhaled breath condensate is a non-invasive biospecimen. Is easily collected from epithelial lining of our respiratory tract. This biospecimen collects a variety of biomolecules like DNA, RNA, microRNA, proteolytic enzymes, etc. So microRNAs are non-small coding RNAs. MicroRNAs are non-small coding RNAs which are regulated in gene expressions. So these microRNAs also plays a crucial role in multiple cellular process. multiple cellular process, and this microRNA also shows to be associated with number of disease including tuberculosis. Axial breath condensate is collected over 10 minutes by an R-tube as per recommended protocol. My aim for this study is to develop a non-invasive diagnostic tool for the early detection of pulmonary tuberculosis using axial breath condensate. As the material and methods, EBC was collected from 10 pulmonary tuberculosis patients and 10 healthy controls. The RNA was extracted by the commercial available kits, and this RNA is used for the cDNA synthesis, followed by the microRNA profiling with the principle of qPCR microarray. Here's the workflow. The demographic details of the recruited patients and controls. Here's the BMI, smoking status, and history of TB. After the data analysis of microarray of pulmonary TB patients and healthy controls, we plot this volcano plot shows 133 microRNAs were differentially expressed and upregulated in pulmonary tuberculosis patients as compared to healthy controls. After the further analysis, we found that 30 microRNAs were 100-fold high in pulmonary tuberculosis patients with the compare of controls, and 3 sets of microRNAs were 1,000-fold high in pulmonary tuberculosis patients as compared to controls. These 3 microRNAs are MID421, MIR1292, and MID92B. These microRNAs also shows in studies that has been involved in respiratory pathways and inflammation immune response. We can say that this differential expression of microRNA may be represented the pathogenesis of the disease in different cell types and inflammation responses. So here we can say that microRNA can be used as a biomarker for the detection of pulmonary tuberculosis using this axial breath condensate. Here's the limitation of my study. This study had been done in less number of sample size, and these differential expressed microRNA we have to validate in the large number of axial breath condensate of tuberculosis patients. Here's the references. Financial support of this study by the AIIMS and ICMR. I would like to acknowledge subjects who have participated in this study and the AIIMS and ICMR.
Thank you. Thank you very much for finishing this talk within very much stipulated time. Sorry. Any question from the floor? Any question from the floor? I will just wait for 5 seconds more. Please. Can you use mic?
So, do bacteria also have microRNA?
Sir,
And are the microRNA you are detecting human or bacterial?
Sir, we are showing the human microRNAs here.
So, human microRNA might be upregulated in many inflammatory conditions.
Yes,
How
sir.
do you prove that these are related to tuberculosis and not other inflammations?
Sir, we are expecting that this sample comes from our respiratory tract, and we are expecting the microRNAs are circulating biomolecules. So we are expecting that these microRNAs are upregulation or downregulation due to the infectious agent. Because we are comparing with controls
So
with
did
no
you-
respiratory disease.
Did you include other pneumonias as control?
Sorry, sir?
Did you include other pneumonias as control?
Not yet, sir. I have to. Yes, sir.
Yes, my comment on this is it is very relevant point that there are other infectious organisms present.
Yes, ma'am.
And if you can relate it to only to tuberculosis, then you can have clinical implications for the diagnosis or other aspects if you can relate it to a higher number of bigger sample size.
Yes,
But
ma'am.
I think in that case you need to increase the sample size as well as you have to increase groups also, so to ensure that there is some kind of specificity and reproducibility also.
Yes, ma'am.
Any other comment?
What is the previous data on this exome in TB?
Sir, there's so many microRNAs have been shown, which is showed downregulated or upregulated in inflammatory responses, which people having tuberculosis disease.
So what is the novelty here?
Sir, novelty here is first of all the sample, exhaled breath collection. So these microRNAs has not been showed yet in EBCs.
Wonderful.
Can you-
I would appreciate your work
Thank you,
because exhaled breath condensate is now being increasingly tested for different infectious, non-infectious disease conditions. So this is excellent work.
Yes.
My just one question is,
Yes, sir.
do you think that even subclinical infections, this microRNA can be detected? B, what would be the critical threshold of microRNA increase when you say that yes, this could be the tuberculosis?
Sir, for the confirmed threshold, we have to do this in a very large number of size. And I would say, like when I do qPCR, I'll take the threshold like more than 18 and less than 35 CT values for that.
Thank you. Oh, sorry, last question.
What about the latent tuberculosis, which is the major burden?
What?
Which is a major burden in latent tuberculosis?
So latent tuber in India, like
Why your sample, you, instead of taking the healthy control, you would have taken another
sir,
round?
I have to. Actually, there's only 10, and I didn't check for the controls, Montox or anything.
Thank you, thank you.
Thank you. so
Thank
much.
you very much. Since this is a pilot study, lots of questions were raised, but I think your detailed study will give answer to all these questions.
Thank you.
Thank
so
you.
much, sir.
Our next presenter is Meenakshi Mendiratta from Department of Stem Cell Facility. She will be talking on dual modification strategies to enhance the targeting efficiency of mesenchymal stem cell derived small extracellular vesicles.
Thank you, ma'am. A very good morning to everyone. I am Meenakshi Mendiratta, second year PhD student under the supervision of Dr. Sujata Monty at Stem Cell Facility. I would like to thank AIIMS for giving me an incredible opportunity for the platform presentation on third annual research day. And the title of my presentation is Dual Modification Strategies to Enhance the Targeting Efficiency of Mesenchymal Stem Cells Derived Small Extracellular Vesicles. Before moving to the results section, I would like to give you a brief background of our study, as our study revolves around liver fibrosis. It is one of the 10th most common cause of death in India, and the major etiology of these diseases are alcohol and the viral infection. And the major cells that affect during liver fibrosis are the hepatocytes and the immune cells. So we are mainly focusing on these two cells, hepatocytes and the immune cells, that is lymphocytes and the macrophages in our study. Under regenerative medicines, several clinical trials have been used MSCs to treat the hepatic disease. And these clinical trials have been used bone marrow and the water jelly in their study. So we are mainly focusing on both these sources, bone marrow and the water jelly. However, there are several limitations on extracellular vesicles derived from mesenchymal stem cells. They are easy to store, more stable than parent cells, and cross blood-brain barrier due to its small size. So, and these MSCs, EVs have been used in various preclinical and the clinical trials to treat the liver disease. And these MSCs, EVs regulate the macrophage polarization, hepatocyte injury, and also regulate T-cell proliferation. Now, however, there are also some limitations of these naive EVs. So to overcome these, some researchers have been used MSCs to modify either its cargo content as well or surface. But in our approach, we modify both its cargo as well as surface content to target the hepatocytes. So these are the strategies that we are adopted to develop the bioengineered EVs to target the hepatocytes. In first, for cargo modifications, we use miRNA-based transfection, and in second, for surface modification, we modify the surface of these EVs with the catenized pullulan which target the hepatocyte receptor, that is ASGPR, acyl-glycoprotein, which is present on the hepatocytes to mitigate the liver disease. And this is the outline of the study. Firstly, we will isolate and characterize these MSCs and EVs as per international guidelines. Coming to the result section, all these MSCs, both bone marrow and water jelly, meet all three criteria. First one, it have a fibroblast-like morphology, differentiate into osteocytes, adipocytes, chondrocyte, and also exhibit more than 95% of MSC-specific marker. And after characterization, we will develop and validate bioengineered EVs by both modify its cargo as well as surface content. So this graph shows that after modify the cargo as well as dual modified SCVs both upregulate the miRNA expression, which reveals that after surface modification, the cargo it does not hinder the process of cargo packaging. So in surface modification, we confirm the presence of catenized pullulan via lectin-induced aggregation method. This graph shows that the turbidity of the solution is increased when bioengineered EVs interact with lectin, which mimics the ASGPR receptor. Now, then we perform the biophysical characterization of naive and bioengineered EVs. In this, the bioengineered EVs also, it maintain all basic characteristics same as naive EVs, but apart from zeta potentials, in which bioengineered EVs has more positive charge, which confirm the presence of catenized pullulan on the surface of bioengineered EVs. Then we perform the functional assessment of bioengineered EVs for oxidative stress elevation in hepatic cells, and results found that in terms of tissue source, water jelly is found to be fare better than bone marrow source. And dual modified SCVs is also significantly reduce the level of oxidative stress than single modified either with cargo or surface. So to validate the target efficiency of these bioengineered AVs towards hepatocyte, we did the same in skin fibroblast cells, which is a non-hepatic cells, and it shows that after surface and dual modified SCVs, it did not elevate the oxidative stress level. So in terms of immune response, we performed the macrophage polarization, and in this we assess the therapeutic potency of these bioengineered EVs to evaluate that it whether it polarized to the pro-inflammatory marker towards anti-inflammatory marker. So we characterize M1 macrophage marker by INOS and M2 marker by CD206 and arginase. And we found that water jelly EVs exhibit superior capability as compared to bone marrow. And it also manifests anti-inflammatory polarization. So the same can be validated by the expression of pro and anti-inflammatory cytokine level. For M1, we perform with IL-1 beta, TNF alpha, and for anti-inflammatory M2 macrophages, IL-10. It also shows the same result. Then next, to assess the potency of these bioengineered EVs to whether it inhibits the T cell proliferation and differentiate towards Treg, then we found that bioengineered WJ SCVs inhibit the T cell proliferation and it differentiate towards T regulatory cells, and thereby it mitigate the inflammation process. And the last one, we perform the same with helper T cells, and it also shows that the bioengineered EV suppress the proliferation of T helper 1 and 17 cells and promote towards T helper 2 phenotype. Then, to sum up this, in terms of tissue sources, water jelly exhibits superior immunomodulatory potentials as compared to bone marrow cells, and bioengineered EVs demonstrated the enhanced targetic efficiency and immunomodulatory and oxidative stress level as compared to the naive EVs. And our in vivo assessment of bioengineered EVs is ongoing, and this work is under patent filing. And our next phase is to perform the transcriptomics of the CLD patients' derived EVs to identify the more targeted miRNAs and the comprehensive multi-omics analysis to gain insight the depth mechanism of action. Now I would like to acknowledge my mentor, Professor Sujata Monti, and all members and funding agencies. Thank you.
Wonderful presentation. Now open for questions. Any question from the benches other than the first three? The row other than the first three. Any question from the students? Such a wonderful work. I think it should raise some questions. Okay, now questions from the first three rows.
So what was the difference in the ratios between the Wharton jelly derived stem cells and the bone marrow derived stem cells? From what you showed, it looked like both were in the range of 4 to 5 for the M1, M2.
Sorry, sir.
How much more was the immunomodulatory effect of one versus the other?
Why Wharton jelly is better than bone marrow?
How much better?
In terms of
So can you tell it in some quantity? Was it 5 times better, twice as better, or both were almost 10-20% of within each other?
Yes, sir. Sir, Wharton jelly is mainly 2 star significant better than the bone marrow in all terms of immunomodulatory and as well as the oxidative stress.
Can you explain that 2 star? What is that?
It's significantly.
No, but
better
how
than-
much is the difference? There can be a significant difference, but
Yes,
the
sir.
difference might be 40% and 50% are also significantly different from each other. So how much more is the Wharton jelly compared to the bone marrow?
It's around 30% better than bone marrow in terms of immunomodulation.
Which statistical test you had used for your experiments?
It's 1-way ANOVA. 1-way ANOVA.
What were the markers of oxidative stress that you have included?
इधर मीनाक्षी इस side में.
What
Sorry.
were the oxidative stress markers that you have included in your study?
Oxidative markers. Ma'am, actually we treat the cells with the H2O2 to induce the ROS level. Then we stain this with the mitosox dye, which measure the ROS level in cells via flow cytometry.
Okay, one.
See, you have mentioned some targeting with the ASG receptor to hepatocyte.
Yes, sir.
So, but with the macrophage, that targeting doesn't work, does it?
No, sir.
So, so your targeted ASG receptor was only specific to your. You've done some hepatocyte specific targeting in that. So, what is the kind of extent of efficiency that you get, the difference?
Okay, sir. So we modified this with catenized pullulan, which mainly target the ASGPR receptor present on hepatocytes. But in macrophages also C-type lectin receptor, which also target of catenized pullulan, and in T cells toll-like receptor 4, which is also target in which catenized pullulan also target this receptor, toll-like 4 receptor on T cells.
You said that you are second year in PhD?
Yes, sir.
A second year PhD student coming on the stage facing 40 years experience professors and responding so efficiently, I think salute to you.
Thank you,
Wonderful.
Thank you,
Just
sir.
my one question, in one of the slide you mentioned that it can be used as an off-label regenerative medicine.
Yes, sir.
What does it mean?
Off-shelves mean the cell-free approach.
Off the shelf, not off-label,
Yes,
off
sir.
the shelf.
Means in the exosome which is derived from MSCs. I mean, it's the cell-free approach in which, due to its small size, it will cross the blood-brain barrier as well as target the specific site of the tissue or organ.
Big clap for her, I think. So you wanted to ask. Just one second.
This is the second year of your PhD. The whole work which you have shown is yours work or somebody else work?
No,
Is it
sir,
very
it's
formal?
my work.
Thank you.
Thank you, ma'am.
The next presentation is by Manvi Arora from Department of Biochemistry. She'll be talking on integrated analysis of expression correlation and signaling pathway connecting FAT1 and EGFR, a WHO-defined molecular marker in astrocytic tumors.
Thank you, everyone. Good morning, everyone. Today I'll be presenting integrated analysis of expression, correlation, and signaling pathways connecting FAT1 with EGFR, a WHO-defined molecular marker in astrocytic tumors. So first we'll define gliomas. So these are the cancers that originate from the glial cells, of which the astrocytic tumors are the most common and aggressive form. So they are classified by the WHO. So earlier they were classified on the basis of histopathological features, but with the advancement there have been inclusion of various molecular markers. So there is a now histopathological diagnosis along with genetic or molecular oriented diagnosis. A recent classification was published in 2021 in which the astrocytic tumors are mainly divided or classified on the basis of IDH mutation status, wherein the astrocytomas are the IDH mutant ones and all the IDH1 wild type have been classified as the glioblastomas. So one important factor is that EGFR amplification and mutation is a common phenomena for the glioblastoma samples and not the astrocytomas. So our gene of interest is a FAT1, so which is a 508 kilodalton type 1 transmembrane protein that can act as a tumor suppressor or oncogene in the context-dependent manner. But in the case of glioma, it acts as an oncogene. So our lab has been identifying the role of FAT1, and we have observed that it's overexpressed in most of the astrocytic tumors, and it regulates the pro-inflammatory microenvironment, migration and invasiveness, EMT and stemness. It suppresses the immune cell infiltration towards the cancer cells, and also it regulates the oncomere to downregulate PDCD4. So we have already demonstrated the oncogenic role of FAT1. So we have here tried to explore the link between FAT1 and the known molecular markers of glioma such as IDH, P53 and EGFR, most common ones. So we have done the tumor sample study where we have analyzed the mutation status, investigated the correlation, and further followed by in vitro studies. So first we have taken up 46 astrocytic tumor samples from the neurosurgery department, and in order to classify them, we have detected the mutation of IDH and P53 using IHC and sequencing. So all the IDH mutants, that were 26, were classified as astrocytomas, and the IDH wild type were classified as glioblastoma samples. And here it is that most of the samples, approximately 55%, had P53 mutation present in them. So then we did the FAT1 expression in these, and we found that approximately 65% of astrocytomas and 55% of glioblastoma samples had upregulated FAT1. So then we did the expression correlation, and we found a positive correlation between FAT1 and IDH and P53. But in the case of EGFR, one of the most interesting finding was that it was only positively regulating with EGFR in the astrocytic samples, astrocytomas, and not in glioblastomas. So we were saying that this might be because of the small sample size. So we did the in silico analysis of the TCGA samples, and we found very interesting finding that FAT1 positively regulated with EGFR only in the astrocytic samples, astrocytomas, and not in glioblastoma. So we were wondering, why does EGFR have a positive correlation only in astrocytomas and not in glioblastomas? Then we remembered that EGFR amplification and EGFR V3 mutation is very common in glioblastoma samples. Thereby we detected them using EGFR amplification was detected using FISH, and V3 mutation was detected using real-time PCR. And we found that 50% of the samples of GBM were showing EGFR amplification, but there was no correlation between FAT1 and EGFR with or without EGFR amplification. And the 33% of samples had EGFR V3 mutation, and there was a positive correlation between FAT1 and wild-type EGFR mRNA expression only in the EGFR3 mutant samples. So as we have seen that EGFR amplification and mutation leads to aberrant EGFR signaling that leads to the increased aggressiveness of gliomas, and our finding that it only positively correlates with FAT1 in astrocytomas and not in glioblastoma samples. So we are we wanted to see, does fat have a role in regulating EGFR in gliomas that are without EGFR amplification? So that might lead to prognosis. So then we defined it, we did the in vitro studies, and we found after fat knockdown using siRNA in 3 different glioma cell lines that there was a significant downregulation of EGFR at RNA as well as protein levels after post-fat knockdown. So we did the promoter analysis of EGFR and we using PromoAlgin software, and we found that there is a transcriptional factor relay which had 5 binding sites at the promoter region of EGFR, and we have also observed in our previous study that fat knockdown down-regulates the expression of relay. So we found that after fat knockdown, there was a significant reduction in the relay expression in 3 different glioma cell lines at RNA and protein level. And we also detected the expression of EGFR after EGFR relay knockdown. And we found post-relay knockdown, there was a significant reduction in the EGFR mRNA as well as protein level post-relay knockdown. So we are here to describe the role of FAT1 in the astrocytic sample. So while we downregulate, we target FAT1, that might lead to reduction of relay, that will lead to further downregulation of EGFR expression, and that might reduce down the glioma progression. So thereby, FAT1 is a promising therapeutic and prognostic target in patients with EGFR expression without EGFR amplification in the case of astrocytic tumors. So here we are trying to basically detect a poor prognostic marker EGFR, and if we are able to regulate by down-regulating FAT1 EGFR, so we can actually reduce down the progression of the disease from lower grades to higher grades, as low FAT1 expressing samples has higher survival rather than high FAT1. So I want to acknowledge Professor Kunjang Chosdol for supervising my work, Professor Subrata Sinha, Professor Partho Prasad Chattopadhyay, Vaishali Suri for doing the diagnosis, for Ashish Suri for providing us these samples, for funding agency, DBT and AIIMS for funding the project, the infrastructure of AIIMS, and the patients specifically, they were really cooperative, the lab members and the staff members of AIIMS. Thank you so much.
The presentation is quite clear, very good. It's open for discussion.
First, from the last benches other than the first 3 rows. The first opportunity for the benches other than the first 3 rows. Any question from? No question from the audience in the first, other than the first 3 rows? Arey, पूछो तो सही. Okay, you don't want to oblige your friend. Now, first 3 rows.
Yeah. So you have worked out very nicely a sequence of events. Just to ask, aside from the areas which you have studied, what is the role of TERT in glioblastoma?
So the role of TERT
in glioblastoma.
Okay, so in the case of glioblastomas, so there is a TERT promoter mutation which is very common in the IDH wild type glioblastoma samples. So it is not with the recent classification that was published in 2021, TERT promoter is not a main molecular markers that has been studied in the astrocytomas, but in the case of glioblastoma, which are IDH wild type samples, TERT promoter mutation is a very common phenomena, and it actually leads to aggressiveness of gliomas.
Can you explain why? Why does it lead to that?
So basically, TERT is the which actually increases the survival. So there is a promoter mutation which will enhance the expression of TERT, and thereby increases the survival of the cancer cells. So there is this end problem of transcription that happens. So that reduces that problem. So thereby it increases the survival of cancer cells.
And the full form of TERT is?
Sir, it's telomerase end transcription factor.
In your experiments,
Sorry, sir?
in your experiments of expression of RNA, how many housekeeping genes have you used as a control?
How many internal? 3 internal controls. We have used 3 internal controls.
So which were they?
So these were 18S transcription, ribosomal RNA, POL, 2A, that is a polymerase, and PPIA, that is an isomerase.
So why did you choose these three among the several housekeeping genes are available?
Yes, sir. So basically what happens in the case of, we can't use, so one of the common internal control is GAP-DH, but in the case of gliomas, there is a hypoxic microenvironment which is present. So thereby GAP-DH is actually regulated in the case of hypoxia, thereby it changes the expression, so we can't use that.
I want to ask you a soft science question. You said patients were very cooperative.
Yes, ma'am.
In scientific term or ethical terms, what does that mean?
So basically what my cooperation of with the patients was when I went to them for taking the ethical clearance. So basically we had to ask them that we will use your sample after post-surgery for a research-oriented process and not for diagnosis per se. We are not basically providing you the clinical report, but we are using it for the research purpose so that we can identify the targets that we can use as a therapeutic basically for medications or anything. And they were very happy to contribute in that study, and that is something I found very impressive about them. So they are really nice.
Good. So that is the underlying current which we have when we do good science to also increase the scientific temper
Yes,
amongst
ma'am.
our
Yes, ma'am.
public at large,
Yes,
and
ma'am.
their contributions to development of newer methods.
Yes, ma'am, definitely.
Yeah, thank you. Very nice presentation.
Thank you,
This TOT promoter mutation, which is not unique to the GBM, it was from my lab we have done TOT promoter in thyroid cancer. And it has been demonstrated very well that it increases the risk of recurrence very high in adult thyroid cancer. But when we did in the children, we found that those patients who have already distant metastasis, that is nothing to do with TOT promoter. So just like P53 once upon a time give some award to Dr. Sinha in late 90s, and that was now everywhere. So this role of this TOT promoter in GBM is not yet established. That's what I wanted to tell you.
Okay,
It's not that you told it has got big role. Its role is not defined. It's not unique to GBM. It is unique to many other cancers.
Yes, sir. Yes, sir, definitely.
Sir, can I? Hello, very good presentation.
Thank you, ma'am.
Just wanted to ask that if you identify this as a biomarker, how do you plan to take it as a translational? Do you plan to develop a commercial assay and take it to the level of a diagnostic kit or something?
So basically what we can do here with this particular study is that we have identified that FAT1 is overexpressed in the case of astrocytic tumors, and it regulates the EGFR. That is a major factor for the progression of a disease from lower grade astrocytomas to higher grade astrocytomas. So when we are targeting glioblastomas, and there is, even after so many therapies, I mean surgery, chemotherapies are available, but still there is no much effect on the increasing the survival of these patients. So thereby when we target FAT1 and EGFR, and even FAT1 which is overexpressed in these samples, we can actually reduce down the progression of a disease, and thereby we can identify an inhibitor.
What I wanted to ask you was, once you have identified this is a biomarker and it can predict progression of disease. So we want an early progression marker to be identified so that patients can get the benefit.
Yes, ma'am.
So how do you take it as a translational, developing a diagnostic kit?
So
Do you plan, could you think of doing that?
Yes, yes, ma'am. Definitely we can, it is a shift from earlier classical diagnostic things to a molecular-oriented diagnosis of things. So FAT1 can be used as a diagnostic marker for astrocytic tumors because it's highly expressed in more than 50 to 60% of the samples, and thereby we can go for a target therapy also.
Thank you.
Thank
Well
you
done.
so much.
All the best.
Thank you so much.
Thank
We
you, ma'am.
conclude this session with this presentation.
Thank you so much.
Thank you, chairpersons. Thank you, judges. Thank you, speakers, for this invigorating session. I now hand over the stage to Dr. Yogita for the next set of platform presentations.
Thank you, Dr. Sanjukta. Good morning and a warm welcome to you all. Now we'll have a session, platform presentations by MBBS students. It's an honor to invite distinguished chairpersons on the stage for this session, Dr. Neena Khanna and Dr. Jayanta Bhattacharya. Dr. Neena Khanna, the former Dean Academics and Head of the Department of Dermatology and Venerology, AIIMS Delhi. Welcome, ma'am. Dr. Jayanta Bhattacharya, current Executive Director and Dean at THSTI from Department of Biotechnology. Welcome, sir. I now request the honored chairpersons to please commence the session.
Good morning. It's always such a pleasure to come back to my MICA, and even more gratifying to see my undergraduate bachas coming on stage, and to steal Dr. Gupta's words when he said that you have these young Turks facing people with 40 plus years of experience, almost sort of waiting to pounce on them. So all the best, and I'm sure these guys are going to do wonders.
So may I invite the first speaker, Ishan Gupta, and the title of the talk is Factors Affecting Recompensation in Hepatitis B, Hepatitis C, and Alcohol-Related Decompensated Cirrhosis, a case control study.
Thank you so much, sir. A very good morning to all present. I'm Ishan Gupta, currently an MBBS intern at AIIMS New Delhi. The topic of my presentation is factors affecting recompensation in hepatitis B, hepatitis C, and alcohol-related decompensated cirrhosis. I would like to begin by talking about the natural clinical course of a patient who presents with a liver injury. In our case, it might be a hepatitis B or C virus or an alcohol-related etiology. So the liver will get, the liver hepatocytes will develop inflammation once the insult starts. They'll gradually progress to fibrosis, cirrhosis, and the cirrhotic liver disease will ensue. Following this, the patient might enter into a stage which we know as the decompensated liver disease by the activity of any of these acute precipitating events in the form of an ascites, hepatic encephalopathy, a GI bleed, or jaundice. We also know that in the case of a cirrhotic liver disease, if appropriate etiology-specific treatments are given and lifestyle modifications are done, the patient can revert histopathologically back and a clinically compensated state. However, nowadays we are talking about after decompensation, the patient returning to a different state. So following decompensation and presentation to the hospital, when the patient is begun on etiology-specific treatment, there can be 3 different disease courses. The patient might progressively decline despite being on therapy. The patient might improve but still remain decompensated, or the patient may enter into a stage which we know as recompensation. So what exactly is hepatic recompensation? So hepatic recompensation, as defined by the Bevino 7 consensus statement, means that the cure of the major etiology of the liver disease, resolution of any of the causes of decompensation, that is no jaundice, ascites, or hepatic encephalopathy, no bleeding event for the past 12 months, and improvement in liver function assessed by bilirubin INR and albumin levels, as well as the patient being off medications for at least 1 year. Previous studies have shown that in viral associated hepatitis, when we give the appropriate antivirals, there is regression or reversal of fibrosis, a decline in the liver stiffness measurements, which can be taken as a proxy indicator for fibrosis, and an improvement in the inflammation of the hepatocytes in about 2/3 patients. In alcohol-related cirrhosis, when there's abstinence, we see a small proportion of patients who go into recompensation. And definitely when a patient undergoes hepatic recompensation, there is a significant risk reduction of mortality. However, in this background, there are certain gaps which I would like to address. There is very little literature as to what factors are associated while determining hepatic recompensation. What is the natural clinical course after a patient achieves hepatic recompensation. So it was in this background that we decided to conduct our study. Our aim was to assess the factors associated with recompensation in decompensated cirrhosis patients. Our objectives were to identify clinical, laboratory, and imaging characteristics associated with recompensation and to determine the prevalence of osteopenia and sarcopenia as measured on CT scans. So moving over to methodology, our study is a retrospective case control study with cases being patients who achieved recompensation and controls being patients who remained decompensated. Patients were followed up until May 2023, and it was conducted at our department, the Department of Gastroenterology, AIIMS, New Delhi. So patients with decompensated cirrhosis with either of the etiologies and in whom etiology-specific treatment was initiated after presentation were included. Patients with dual etiology or an HIV co-infection or other etiologies were excluded. We collected data pertaining to clinical and anthropometric parameters, pertaining to transient elastography through FibroScan results, and child-pug and mild, that is model for end-stage liver disease scores for liver function. We also did CT abdomen for checking for osteopenia and sarcopenia. Here we can see an osteopenic vertebra with a mean score of 116 Hounsfield units because the cutoff is at 149 Hounsfield units. This is a non-osteopenic vertebra, and these are scans at the third lumbar vertebral level for measuring the skeletal muscle index for calculating sarcopenia. Moving on to results, our patients were 109 in number, with 39 related to alcohol-related etiology, 35 each for hepatitis B and C. 40 patients achieved recompensation, while 69 remained decompensated, and the median duration of follow-up was 2.8 years. These are the baseline clinical and laboratory parameters. As you can see that the age at index decompensation, the baseline MELD score, and the serial change in the liver stiffness measurements, particularly the decline in liver stiffness measurements, was seen to be significantly associated with predicting recompensation. This is a plot depicting the earliest MELD score versus the MELD score at latest follow-up. As you can see, not only those who recompensated had a lower baseline MELD score, those who had a higher MELD score at initial follow-up had a significant decline in their MELD scores. This is a plot depicting the liver stiffness measurement scores, and we can see that in the recompensated cohort, we can see a decline in the liver stiffness measurements on last follow-up, which was also statistically significant in our results. We also did univariate analysis, but for which the age at index decompensation and one of the etiologies came out to be statistically significant. However, because of the sample size, we were not able to do a multivariate analysis. So I would like to conclude by saying that the lower age at index decompensation, lower MELD score at baseline, and a greater decline in liver stiffness measurements on follow-up were associated with recompensation in our study. However, sarcopenia, osteopenia, or the time to treatment initiation were not associated with recompensation. There were certain limitations in our study. Our study was retrospective, so we could not assess all the parameters. Definitely the patients who recompensated had a higher survivability, so that introduced a bias. The sample size was relatively smaller, and the findings were only valid for hepatitis B, C, and alcohol-related etiologies, because for these etiologies, we have the etiology-specific treatment available, antivirals and the alcohol abstinence. And we are actively recruiting patients. We have already done more than 140, 150 patients at the moment, and we plan that after the end of this study, we aim to develop a model which will help us in categorizing patients based on the need for transplant, so that we can develop an algorithm to categorize these patients based on their needs. Thank you.
Thank you, Ishan. Can we have questions first from the house? You've not called any of your classmates and tutored them what questions to ask? And that's what normally people do. Okay, can we have the first 2 rows, 3 rows?
Excellent presentation, Ishan. Many of the decompensated patients have ascites, right? So could that have affected your transient elastography results? Because ascites is the confounding factor when you do TE.
So as a part of our definition for recompensation, we take recompensation only when there is resolution of ascites, jaundice.
No, but your baseline TE was done at when the patient decompensated.
That is true.
And that time he could have had ascites, right?
That could have compounded our results, yeah. I think we need more sample sizes in both our cohorts, but I feel that still there was an increase in the decompensated cohort. So there was some plus in a single digit, and there was a decrease in 20 units in kilopascals in the recompensated cohort. So I think even if there were few confounders, I still feel that this result was significant. Again, if we include more patients, we'll definitely have more confidence in this.
Any questions? Are there any other questions?
Hello. Yeah, nice presentation. So a basic question regarding your this thing. On the basis of the etiology, whether HB B or C or the alcohol intake, was there a difference in the decompensated level which has affected the outcome?
So we did not evaluate it separately based on etiologies, but what we see is based on all of these etiologies, histopathologically and even clinically, we don't see a difference in these patients. So the cirrhotic liver disease or the mode of decompensation. So if I say there are 4 modes of decompensation, there is no clinical difference in these etiologies, and even on histopathology, there's no difference. So there might be, we have not done that subgroup analysis.
Okay, thank you.
Any other questions? Okay, thank you, Ishan, very well done.
Thank you.
Our next presentation is by Akansha Singh. She's going to be speaking on non-invasive assessment of dysautonomia in COVID-19 survivors using short-term heat HRV.
Hello everyone, I am Akanksha, currently a third year MBBS student here at AIIMS, and today I'll be presenting my research topic, which is non-invasive assessment of dysautonomia in COVID-19 survivors using short-term heart rate variability. I have done this along with the mentorship of Dr. Deenu S. Chandran in the Department of Physiology. So, the background. It is seen that many patients, after their complete recovery from the acute phase of COVID-19, developed many debilitating symptoms, such as orthostatic intolerance, dizziness, fatigue, palpitations, and many other system ailments developed across them. And this was termed the long COVID or post-acute COVID-19 syndrome. Various studies were conducted, and this suggested that COVID-19 infection affects the autonomic nervous system, causing dysautonomia, which could be responsible for some of these changes. So, to assess this dysautonomia, the aim of our study is to assess this dysautonomia using short-term heart rate variability in COVID-19 survivors and compare it with age and gender-matched historical control data. The study is a cross-sectional observational study performed in the Autonomic Function Laboratory here at AIIMS, Delhi. So for the study, we recruited 44 COVID-19 survivors who had totally recovered after 28 days and within 3 months of their recovery. And for the controls, we took historical controls because most of the studies which were done in this field took controls from within the COVID-19 period. And as we know that the prevalence of subclinical infection was a lot, so there could have been some missed cases and that could have hampered the result. So to eliminate this thing, we took historical controls from before the pre-COVID era, that is before January 2020. Now, for these 88 subjects, the 44 COVID-19 survivors and the 44 controls, we took their 5-minute ECG recording. It was acquired using Power Lab 4x26, and then it was analyzed to extract the RR intervals using the software LabChart Pro. After that, a manual editing was done to remove all the ectopic beats, the false beats, and further the results were obtained that were called NN intervals. Okay, so, and after that, these NN intervals were subjected to QBOS software to analyze 3 domain parameters, the time domain parameters, the frequency domain parameters, and the non-linear parameters. So now let us understand what exactly is HRV. HRV is the variation or the fluctuation in the RR intervals along successive heartbeats. Now why is it occurring? It happens because of the autonomic nervous system. We know that the autonomic nervous system impacts our SA node and hence leads to these variations in the heart rate. So more is the fluctuation, more is the modulation
in which the SNS and the parasympathetic system can act, the better the outcome will be, because we need our autonomic nervous system to function in case of stressors, okay? So the more it is, the more is the HRV, the better cardiovascular health it is. So we want it to be more. So as you can see, what we did was, these parameters which were obtained, the time, frequency, and the nonlinear parameters, they were used to assess how much is the heart rate variability. And now I would like to tell you a few important points about how the sympathetic and vagal influence acts. The sympathetic influence acts via adenylate cyclase enzyme, and the parasympathetic influence, it acts via G proteins, and it directly acts on the potassium channels. So since we know that adenylate cyclase is an enzyme, it takes a bit more time. So the low frequency changes which occur in heart rate are mediated mainly by sympathetic influence, and the high frequency changes which occur are mainly mediated by vagal influence. This will be important in understanding the further results. Okay, so now with this, let's come to the results. So in the time domain parameters, the two important parameters which we assessed were SDNN and RMSSD. SDNN is the standard deviation of NN intervals. The RMSSD is the root mean square of difference of NN intervals. Now both these parameters, as we can see, it was reduced in the COVID-19 people. That means the modulation was actually reduced. Now let us come to the frequency domain parameters. So to obtain the frequency domain parameters, what we did was, we plotted those RR intervals on y-axis along with the time on the x-axis, and then this was passed through Fourier transform, and the Fourier transform actually splits it into low frequency data and high frequency data, and how much is the spectral power in those frequencies is then assessed. So as I discussed earlier, that the low frequency corresponds to sympathetic influence, and the high frequency corresponds to parasympathetic influence. And as we can see, both the LF and HF absolute powers were actually reduced in COVID-19. Now let us come to nonlinear parameters. So nonlinear parameters are SD1 and SD2. SD1 is actually, as you can see, SD1 is the standard deviation along the transverse axis when we plot the RR interval n and RR interval n plus 1 on the y-axis, and SD2 is the standard deviation along the line of identity. The SD1 here corresponds to instantaneous beat to beat variability / vagal influence, and the RMSSD and the SD2 corresponds to sympathetic influence. And both of these were significantly lower in the COVID-19 group. So from here, we can see that our results points towards significant impairment of autonomic function in COVID, and this has been in correspondence with many other studies like Menezes et al., Liviero et al., but in contrary, we have obtained other results also which were opposite to this. Now, when we did the ROL, we found out that there could be many reasons which could be responsible for these fluctuations. One could be the neurotropic nature of the COVID-19 virus. Another important thing which came out was performed in our own laboratory here itself at AIIMS, which showed that it could be due to the mechanical impact of arterial stiffening and also reduced baroreflex sensitivity, which was caused by COVID virus. So all these things could lead to decreased ANS modulation. Now it is very important because this serves as physiological alarms of the risk of sudden cardiac death in COVID-19 patients. If a person has reduced heart rate variability, it means their cardiovascular system is not functioning properly, and hence it provides us with an opportunity to screen patients who presents with postural hypotension or postural dizziness, and we can screen them time to time and prevent their cardiovascular mortality. Thank you. I would like to thank Dr. Deenu S. Chandran, without whom this project would not have been possible. Thank you so much, sir.
Excellent talk. Questions? As usual, from the back, you expected first? I mean, I think we all have gone through the COVID phase. I think you should ask some questions, and we all know long COVID, right? Even if you don't have probably understood this, the scientific terms, but you must have been familiar with what is being talked often nowadays. It's called long COVID, right? And she has touched upon some of these aspects. Any questions from the back?
Sir, can I?
Yeah, please.
Yes, very good work, Akanksha.
Thank you so
much.
much.
But just to set the record straight, Dr. Deenu has helped you as a mentor in this, but the conception of the idea, our mentor, Professor Deepak, is sitting there. Sir, 1 minute. Yes. So, who was initiated the idea?
Okay.
project got from ICMR, also the running of the autonomous lab. So myself, Dr. Ashok Jaryal, Professor Deepak, of course, as our mentor, right, and the vascular function lab. And so therefore, I just wanted to give credit where it is due. That is part of our teaching to you also.
Okay.
So congratulations, Dinu, for having guided this work. So that's
Thank you so much.
Sure, please.
and also, thanks for bringing physiology to the forefront of how we can help in clinics. Thank you very much.
Thank you
we are
so
very
much, ma'am.
passionate about physiology, especially at the clinical physiology interface. We have done a lot of things in bench and translational, but this is good. Thank you.
Great.
Thank you so much, ma'am.
Yeah, thank you. It was excellent work, and I just want to, you may not like to answer, that's fine. What is this Fourier theorem on the basis of which all this analysis is done? What is Fourier theorem? Can you answer that? Just for your assessing your knowledge about the work you are doing.
So I don't exactly know what it is. I have compared it with like a prism through which if you pass white light, it splits it. So I just compared it, but I don't exactly know how it operates.
Any repetitive function can be analyzed in form of sine and cosine. That is called Fourier theorem. And that based on, you can change from time domain to frequency domain, from frequency domain to time domain. All this ECG, EEG, everything which is bioelectricity, which is frequency with time, cyclical, you can convert them into the Fourier theorem. Whole based on that, MR imaging, all other imaging, lot of imaging based on Fourier transformation. That is the basis of this work is Fourier transformation. That's why you should have asked your teacher what is Fourier theorem on basis which I am doing spectral analysis.
Yeah, please. After.
Hello, Akanksha, nice presentation and good piece of work. So the patient you have recruited is within 12 weeks of COVID infection. So these are the findings, say within 3 months. So is there any plan to see whether the effect is lasting in these patients maybe after now? Was any study carried out, you know, on these patients after 6 months or 1 year or 2 years of infection to see the long-term effect?
Okay,
Thank you.
ma'am, so we took patients from within 3 months, and it has been found out that most of the effects of long COVID subside by the after 6 months of infection. So it has been suggested that their screening, if they are developing any postural dizziness or any symptoms of long COVID, they get tested after 6 months, and it is probable that most of it will get subsided. If it still is happening or if it still recurs, then at 3 monthly basis, they can be assessed further on. But I exactly don't know what the other studies which has been done.
Thank you.
One more, just one more. This last question.
Yeah. Yes, here. Yes, Akansha, good work.
Thank you.
And Professor Deepak is sitting here, so I am tempted to ask one question at least. In one of the slides, you said that it is because of the changes of this BRS. And perhaps you said the cause is the COVID-19 virus. That may be. But in my opinion, the main cause appears to be stress. The era of COVID-19 itself was a stress.
Okay.
What do you say? Stresses or the COVID-19 virus per se?
Yeah, that definitely can be a variable which could lead to those changes. But stress has been there before COVID-19 phase as well. But yeah, it can definitely be.
Stress is even at this auditorium, we have stress. Everyone has stress.
So, yeah, definitely that can be a reason.
Okay, we need to move on.
Thank you.
We have to move on. Yeah, so excellent talk again.
We'll let you go, Akansha. I think, yes, we have the next.
Yeah.
Thank you, sir.
The next speaker is Rutul Shag, and she's going to talk on risk-taking behavior in young adults with substance use disorder.
Good morning, everyone. I am Rutul Chag, going to present our research work on the topic risk-taking behavior in young adults with substance use disorder. My mentors are Dr. Simran Kaur, Chaitanya Leon, Dr. Ravneet Kaur, Dr. Yatanpal Singh Balhara, and Dr. Ratna Sharma. So, what is risk-taking? Risk-taking behavior encompasses the behavior which are done under uncertainty and without robust contingency planning and may frequently lead to negative consequences. While addiction is defined as use of any substance or engagement in any behavior, and despite its negative consequences, it has a prevalence of 4.6%, especially high in adolescent and young age, which might be due to biological changes, peer pressure, environmental exposure, cultural and family influences. So why risk-taking behavior is seen in young adults with substance use disorder? There is a dual model which explains the same. Substance use disorder leads to higher level of thirst of seeking rewards, which in turn leads to higher impulsiveness. At the same time, it impairs the cognitive control system, which leads to decrease in self-control and decision-making. This together leads to increase in risk-taking behavior. Thus, we hypothesize that higher risk-taking behavior will be present in patients with addictive disorder compared to match healthy control. So, what are the lacunae in previous literature? Previous literature have either studied on alcohol or opioid, but not both together at the same time. They have used either objective parameter or subjective parameter, but not both together at the same time. Also, studies related to risk-taking behavior and opioid addiction are very few. The aim of our research is to study the risk-taking behavior among alcohol and opioid use disorder subject with the objective of first to study and compare the risk-taking behavior in patient of alcohol and opioid addictive disorder as compared to match healthy control using BART and questionnaire. Second, to correlate risk-taking behavior scores with reaction time and accuracy of cognitive tasks and scores of self-control of patients. It is a cross-sectional observational study. Inclusion criteria is diagnosis by the DSM-5 criteria for the respective addiction, and rest of the inclusion criteria are same for all the subject group, which are aged between 16 to 40 years, either gender, should be able to give written consent form and should be able to perform cognitive tasks, and exclusion criteria being the history of neuropsychiatric or neurodegenerative disorder other than addiction. We first obtained the ethical clearance. Then we recruited 54 subjects, 18 each of opioid use disorder, 18 of alcohol addictive disorder, both recruited from the NDTTC Ghaziabad, while 18 healthy controls were recruited from Dakshin Puri community. Then we asked them to perform BART, objective cognitive task, and 3 questionnaire, risk-taking questionnaire, RT18, BSCS, brief self-control scale, and EIS, Isaac impulsivity scale. Then we analyze the data using GraphPad, Prism, V10 software. Coming to the same model, we are going to measure impulsiveness using EIS questionnaire, cognitive control system using BART, self-control using BSCS, and risk-taking behavior through RT18. What is a BART? It is a computerized task that simulates real-world decision-making, balancing potential gain and losses. We use inbuilt software in the PsychoPy. So on BART, at each and every step, person has option either to pump the balloon or he can bank to collect the accumulated money. So let's say he has pumped the balloon till $20 and he want to bank the money. So he has banked the money and he got the money, and total money will be shown at the end of the at the end of the 60 trial. In the second case, let's say he has pumped the balloon till $15, and when next time he has pumped the balloon got burst. There is a probability of bursting the balloon at each and every step, and person doesn't know the probability of bursting the balloon. And in this case, he hasn't gained any money. Outcome that we are going, variable that we are going to use for our research are: first, average adjusted pump, that is average of pump on the banked balloon. Second, average of reaction time on all pump. Third, average of reaction time on safe bed, that is banked balloon. These are the questionnaire. Coming to the result, we are going to present the result under the following headings: demographic profile. All the 3 groups are age and gender matched. Education status is of minimum 10 years. Daily dose, years of consumption, and medication are as shown. Coming to the result of BART, opioid addicted group has shown a higher level of average adjusted pump as compared to alcohol addicted group. They have also showed higher level of average safe bed compared to alcohol addicted group. Opioid addicted group also has shown higher level of average reaction time and average reaction time on the safe bed. Coming to the result of the questionnaire, opioid addictive group has shown higher level of RT18 score, lower level of BSCS score, and higher level of impulsiveness on EIS scale, while the opioid addictive group didn't show any significant difference. On correlation analysis, there was a negative correlation for all the subject group between impulsiveness and BSCS score, that is self-control. Coming to the discussion, There are previous literatures which have done research on both alcohol and opioid addiction, and they have found a positive correlation between addiction and BART score. And they have proposed that BART is a comprehensive risk-taking assessment tool used in addictive disorder. While Campbell et al. Found a negative correlation between BART score and alcohol addiction, and they proposed that it is due to neuropsychological impairment seen in the domains of immediate memory and executive functioning. While there are many research which have found no significant difference, which might be due to limited sample size, age, or methodological differences. Coming to the discussion of questionnaire, there are previous literature which have found the similar result for various questionnaire. There were researches which have also done research on women, and there are researches which have also done longitudinal analysis beside the cross-sectional analysis. Stamets et al. Found a negative correlation between RT18 score and alcohol addiction, and they have suggested that it might be due to binge use frequency, because they have used binge use frequency instead of binge use status, user status, so that's why they have found a negative correlation. Conclusion. We concluded that young adults with opioid addictive disorder displayed higher level of risk-taking behavior as compared to subject with alcohol addictive disorder and healthy control, based on both subjective and objective parameters. Limitation. It is a pilot study with limited sample size with only men's. Future direction. We can use neurological imaging like MRI, EEG for neural correlates of risk-taking behavior. Second, we can prevent the negative outcome associated with risk-taking behavior. Identifying individuals with higher risk can help us to strategize early intervention to reduce the risk-taking behavior. Also, there are literatures which have suggested that higher level of addiction lead to higher risk-taking behavior, and higher risk-taking behavior leads to higher addiction. So they are stuck in a vicious cycle. We would like to thank you ICMR for funding our project. Thank you, everyone.
Thank you, Rutul. Shortage of time, we'll restrict the questions to the judges, please.
I didn't want to ask anything, but I wanted to only
Sir, please.
ask you, why dollar in the balloon? Why not INR?
So it was a pre-built, it was pre-built in PsychoPy, and I don't know the coding.
Yeah, Subrata.
I mean, why is there a difference of is there a difference of personalities between the people who tend to be opioid addicted versus alcohol addiction, that you see this difference between two types of addiction? And what's your hypothesis?
So actually, we haven't found any such literatures which have found similar result, like comparison between alcohol and opioid. There were studies which have found correlation between cocaine use and opioid use. Our result could be might of limited sample size, and we would like to complete the sample size till 30, and hope so ki, we can find the.
Don't hope so.
Okay,
New
sorry,
results
sir.
might be better. I mean, different results might be more important.
Yes,
you know?
and also they are on detoxification medication, so we don't know the impact of the medication also.
Okay, thank you. The judges?
So with the permission of chair, can I ask question?
Yeah.
Can I ask
Yeah,
question?
please.
Yeah, so when we did this study first, we called ODS. How come it has become a different name now? So you may answer or not answer, but why is there a change in nomenclature? It was ODS, opiate dependence syndrome, and we did carry out studies. So that's a question to you, but if it's okay. Why the name has been changed over 30 years? Anyway, the second question, you are actually hitting the target. If you go to the amygdala by neuroimaging, you might find the reward and punishment areas. And when we did this study, we had a real difficulty. We used to take patients from Deen Dayal Upadhyay Hospital, where this was, we never had an ETC here, and patient used to run away on the way. It was very difficult. They were locked. And so the risk taking is enormous in opioid use syndrome or dependence syndrome, whatever you call it. So if you try to do MRI studies and you can hit the target, and then you deal the target by some neuro influencing by non-invasive technique, you are on the right path. All the best. Thank you.
Thank you, sir.
Very well done. Any other questions? Right, thank you so much.
Thank you, everyone.
We thank the distinguished chairpersons and the speakers and the judges for an interesting session. I now hand over to Dr. Kamal Gulati for announcing the next session.
Thank you, Dr. Yogita. A very good morning. Now we move to the keynote address of the day. I request Dr. N.K. Mehra and Dr. Subrata Sinha to kindly come on the stage to chair this session. Professor N.K. Mehra has been former Dean Research and Head of the Department of Transplant Immunology and Immunogenetics at AIIMS New Delhi. And Professor Subrata Sinha has been the former Dean Academics, Dean Research, and Head of the Department of Biochemistry at AIIMS New Delhi. Professor Sinha has also been the Director of National Brain Research Center and is currently J.C. Bose National Fellow. I also invite Professor Shubha Tole to kindly join them on the stage.
Without wasting time, Kamal.
Thank you, sir.
Welcome to the first keynote lecture. And we have a very distinguished speaker, Dr. Shubha Tole. I told her that I have reviewed your CV in the past. I won't tell you where, but I mean somewhere. Very impressive. A lot of her CV is on the slide here. She has actually changed her title, not building the hippocampus, because when I read this title, I thought she's going to talk about stem cells and all that, building the hippocampus, preparing it in the lab. So she's going to talk about 2 genes and 2 mothers, bracket cells. So she's actually going to be talking something on those lines. Shubha is an Indian neuroscientist, professor and principal investigator at the Tata Institute of Fundamental Research in Mumbai. Her research investigations and development and evolution of the mammalian brain. Tole has received several awards. I think she's one of the very highly decorated scientists of India. 2010, she had the Shanti Bhatnagar Award. All of this award has been changed now to a new sort of avatar, but S.S. Bhatnagar Award remains the highest award for science in India. She also had the Swaran Jayanti Fellowship from the Department of Science and Technology, Wellcome Trust Senior in International Fellowship, National Women Bioscientist Award from the DBT Research. These are a few. And she is also the past chair of the Women in Science panel of the IAS International Award for Mentorship for Society for Neuroscience. Shubha, the floor is yours. We don't want to stand between you. We want to know how you talk about your 2 genes and 2 mother cells.
Hello, everyone. Some of you are for the first time. Some of you, we had an amazing time yesterday. And you all know why I'm not standing behind this. There we go. Thank you. Assistive device. Okay, here we go. Now I can see. I'm going to tell you a story about mother cells. I was thinking I would call this a story of 2 mothers. And then I realized that it's actually going to be the same mother cell that changes after it produces one progeny and then the next. Okay, you can anthropomorphize this as you wish, but we are talking about brain stem cells today. This is my beautiful institute, the Tata Institute of Fundamental Research in Mumbai. Lovely campus. And those of us who work on neurons here, so let me introduce the 2 genes actually. They may be familiar to you, FOXG1 mutations of which cause FOXG1 syndrome in humans, part of the autism spectrum disorder, and LHX2, which has recently been reported to cause a series of other types of brain disorders, and I will highlight some of them. So these are the two genes, and what we're going to talk about is how they control what type of daughter cell each mother cell will produce. Okay, that's the name of the game today. And so when neuroscientists walk about a beautiful campus like this, you know, we see trees and we see arbors and we think, oh, neuronal arbors look just like trees. But the brain has more than just neurons. The brain has a vast array of glial cells, in fact, more glial cells than neurons. And then when you begin to work on glial cells, you actually begin to see glia among the trees, like so. See? Yep, you don't see it until you see it. Yeah, so these glia are actually our data, and I thought they fit in so beautifully amongst the trees that, that we could create a collage. So okay, here goes. The first gene I'm going to talk about is FOXG1. What's FOXG1 syndrome? Okay, it includes a devastating phenotype in the brain and in haploinsufficiency. Okay, gain and loss of function can cause many problems. Reduced folds on the surface of the brain, intellectual disability, inconsolable crying in the first year of life. Okay, loss of language, an array of really profound deficits. Okay, males and females, it's an autosomal dominant condition, and somehow this gene seems to be a hotspot for new mutations. Okay, 120 different mutations of an array of types have been found. And there is actually a FOXG1 Foundation that supports and collates research and provides help to families. We began looking at FOXG1 because there is an excellent mouse model for studying FOXG1 mutations, and we discovered a new phenotype that had been previously unreported. This work is the thesis work of my student Mahima Bose, whom I will introduce soon. But let me introduce the system and the mother cells. So one fantastic feature of the vertebrate central nervous system is that common progenitors first produce neurons and then produce glia. So it's the same progenitor that will produce a copy of itself and a neuron, a copy of itself and a neuron, and then it will switch to begin to produce glia, here astrocytes. Okay? It's the same mother cell that undergoes the neuron-glia cell fate switch. And it is this mechanism that we were privileged to discover how it is modulated. How does the mother cell change its gene regulation in order to now produce a completely different type of daughter? So that will be the first part of my talk. We're going to change the embryonic day 15 mother cell. What is this? Ours is a mouse model. Mouse has a 19.5-day gestation. The brain, the neural tube closes around day 8.5, and then the cerebral cortex begins to populate with neurons. So much of the cerebral cortex is born between days 11 and day 16, and it's at the cusp of this, when it is still producing the last wave of neurons, that we're going to talk about the first mother cell. Now, normally the neuron-glia cell fate switch would begin to occur a little bit later. And in the second part of my talk, we're going to talk about a later mother cell that only knows to form glia. Okay, so those are the two mother cells. And here is Mahima. The first part of my work, the FOXG1, is her work. She's recently submitted her thesis, and this work is in review in PNAS. And here is the team that Mahima worked with. Varun Suresh, a PhD student who's graduated and is doing a postdoc at UCSF. Urvi Mishra, a master's student who is now at Boston University doing her PhD. Ishita Talwar, an end-stage master's student who's contributed wonderfully to this project. And here now is a section of the mouse brain. I know you all are used to looking at human brain, but you know what? Early in embryogenesis, the human and mouse brain don't look that different. They all have a relatively smooth cortical primordium. Here's the ventricle. So this is only one half of the brain. This is the midline. Here's where the hippocampus will form, no longer the subject of what I'm going to talk about today. That was a whole different talk, how to make the hippocampus. But I thought you folks would be more interested in this one. So here is FOXG1 in green, and you can see how it is expressed in these post-mitotic cortical neurons, okay? And it's also expressed in these progenitors. So they are yellow because it's a double stain for two different molecules, but so FOXG1 is here in the progenitors. And what Mahima did is she knocked out the FOXG1 gene using a beautiful surgical technique called in utero electroporation. So we obtained the FOXG1 conditional knockout mouse from Gord Fischel and Goichi Miyoshi, who made it. So the FOXG1 locus is flanked by LOXP sites, and when you introduce Cre recombinase, the gene gets deleted. And Cre recombinase is introduced by in utero electroporation. What is that? So here is mouse surgery. Okay, okay, here. Here is the mouse under deep anesthesia. You can, I think I will need a new pointer. Till then I will use this. Yeah. So here's the mouse, that's the nose end here. It's lying on its back, two little paws, and you can see here the uterus is exposed, and each little ball is an embryo. A mouse can have up to 15 embryos. Okay, it's in each little ball. And what's happening, new pointer would be nice, thanks. What's happening here is, now I've lost, is that this pipette is injecting in plasmid DNA, which encodes Cre recombinase together with the GFP. And the other black thing is only a light source to help us illuminate the embryo by transillumination so we can see where the ventricle is. Now the DNA is in, and as a next step, electric current is applied to the embryo from the outside the uterine walls, and this achieves transfection of the plasmid DNA into the cells that are lining the ventricle. So only the cells that are along the ventricle, here the progenitors, they will take up the DNA, and not all of them will, only some will. So what we get is a beautiful mosaic system where the whole brain is wild type, normal, except the progenitors, the mother cells that have picked up the plasmid DNA. Okay, so only some of them will pick up the plasmid DNA and become knockout, and then all of their progeny will continue to be FOXG1 knockout. So we can ask, what do FOXG1 knockout cells look like amidst a normal wild type background? It's a beautiful mosaic system. And this is a little cartoon that essentially shows the same thing. DNA is injected in and electric current applied. And what we do is that we wait for a couple of weeks after this perturbation, because the mother cell has to form whatever daughters it's going to form. Those daughters have to migrate wherever they have to go and differentiate, and then we look at them with markers. So that's the study. Okay, what we found, long story short, loss of FOXG1 gives rise to premature astrogliogenesis. This means a progenitor that was supposed to make a neuron now makes a glia. And this is the little cartoon for that. Okay, we did the electroporation EP at E15, embryonic day 15, and we're looking at postnatal day 14, so more than 2 weeks after. And here's control. So in the control, the green cells are all normal because we did not, so here in the control, the FOXG1 isn't heterozygous, so it's LOX over plus, so there's one good copy of FOXG1. And in this system, haploinsufficiency does not cause a phenotype. And you can see 14 days later, up here you see many dots. These are all green nu-N positive neurons. You can see how the nu-N and the green co-localize, which means all these green cells are neurons. You also see some big fat fluffy astrocytes, because remember the progenitor that is making neurons is later on going to make glia. So you also see green glia, and that's normal, but overwhelmingly the electroporated cells are neurons. What happens when you remove FOXG1? Complete change. You don't see any neurons. You don't see even one neuron. Every green cell that has come out of a FOXG1 knockout progenitor has now formed an astrocyte. And you can see they don't co-localize with nu-N, but instead co-localize with an astrocyte marker, ALDH1L1. And here are the counts. So essentially, the number of nu-N positive neurons in the control So this is electroporated cells. So as a nu n positive on total electroporated cells is this much. And look, not a single neuron in the FOXT1 loss of function, LOF. And the glia correspondingly increase. We've pretty much shown loss of FOXT1, this one gene, is enough to convert a neurogenic mother cell into an astrogliogenic one. Okay? How? How? And here is where Mahima's sheer insight came in. Mahima worked with Varun during the pandemic when experiments were not possible. The animal house was closed. And they embarked on a bioinformatic exploration. What they did is this. FOXT1 ChIP-seq data was known. It was in the database from Bin Chen's lab. So they looked at all genes that were occupied by FOXT1 bound genes. Okay? They also had at their hands, from Ben Barris's lab, a database of all genes that are enriched in astrocytes here. And they overlapped those, okay, which gave a subset. And then they said, okay, neural stem cells, okay, NSCs, the database of chromatin epigenetic marks in neural stem cells was also available. And they said, let us curate this, let us mine this database for genes that are bivalently marked. And what's bivalently marked? There are chromatin modifications that mark active genes, and there are chromatin modifications that mark genes that are silenced. Bivalent means that if a gene has both marks, it's sort of like putting your foot on the brake and the accelerator at the same time in a car. Somehow, bivalent marking makes a gene poised and much more ready to either be on or be off by simply removing the opposite mark. So bivalent genes are considered to be those that are raring to go or not go. And so they thought that in the subset of astrocyte-enriched genes which are occupied by FOXT1, which are bivalently marked, those might be the operational ones in the progenitor that switch the progenitor from neuron to glia. Okay? And this they did entirely informatically during the pandemic because this was what they could do. So it just shows a creative mind can pursue science no matter what the restrictions. And having made this list of little more than a dozen, maybe 20-ish genes, what they did was then subset it when they could do an experiment with FOXG1, with RNA-seq from FOXG1 control and mutant. So essentially they sorted out cells that were electroporated and mutant for FOXG1 and then did an RNA-seq and compared that, and that's just a volcano plot of the known gliogenic targets that got upregulated and so on. And having subsetted it this way, what they got is that this entire list overlapped with the genes that were upregulated in the FOXG1 loss of function, gave 4. So this entire set, vast database, came down to 4 genes. Okay, I still don't have a pointer that's working, so I'll try and do this. Okay, came down to 4 genes, and here is where things got really exciting, because FGF receptor 3, okay, FGF receptor 3, FGF, fibroblast growth factor, is a known potent gliogenic molecule. Literature in the field has shown that if you add FGF to cultures or in vivo, you will convert neurogenic cells to gliogenic. Okay? FGFR3 has now gone up. So the model then is that FOXG1 normally in a mother cell suppresses FGF receptor. When FOXG1 is lost, FGF receptor goes up, the cell now can sense endogenous FGFs and now becomes gliogenic. So here's a beautiful molecular model. Is this indeed the case? Okay, this is FOXG1 occupying the FGFR3 locus. This is from the ChIP-seq data set. Okay, is it really the case? Well, it appears to be so indeed. So in green is FGFR3, in red is AI9. Here, the mutation is achieved not by sparse electroporation, but by using an HGFAP Cree driver that drives the loss in all of the mother cells. So this is why all of the mother cells lining the ventricle are red. This is just an AI9 red fluorescent TD tomato reporter. In control, they're all red, but look, none of them are really expressing high levels of FGFR3 in control. In the mutant, however, look, all of them have turned yellow because it's an overlap for red and green. And all of them have turned on FGFR3. This is sort of just visually, but what Mahima did is she quantitated the amount of FGFR3 in these, and you can see the FGFR3 levels have gone up. Okay, FGFR3 has gone up, but kaam ki baat, is the cell experiencing FGF signaling? Because only that will turn a neurogenic cell to a gliogenic cell. And what they did was they looked at a readout of phospho-ERK, okay? And nuclear phospho-ERK, excuse me, these circles actually represent scoring for nuclear phospho-ERK, not FGFR3. You can see phospho-ERK has gone up. As many of you may know, the FGF signaling upregulates the ERK, the MAP kinase ERK pathway. And so we have confirmation that inside the mother cell, the FGF signaling pathway has indeed gone up, and that is how the mother cell becomes gliogenic. Okay? What if you take away endogenous FGF signaling? What if you remove FGF? And to do together with FOXG1 removal. So FOXG1 removal is making the cell, mother cell, gliogenic, but by using the FGF pathway. So what if you mop up the FGF ligands around by using a construct that is known to do this? It's a secreted FGF receptor that just binds up all the FGF around. Then what happens? Does the cell become back a neurogenic cell? Okay? So this is the last bit of data in this segment. Here's control. You've seen this before, many neurons. Here's FOXG1 loss, many, many astrocytes. And here's FOXG1 loss plus the FGF secreted receptor. You still don't get back neurons, but you see now these many, many tiny cells. So you've not made the progenitor gliogenic, but now you're producing these other cells. What are they? Turns out, okay, here's a high mag, control mostly neurons, FOXG1 loss mostly astrocytes, but together with removing FGF, you now get oligodendrocyte precursor cells, the next type of glial cell that the progenitor will produce. Okay, olig-2 PDGFR alpha forming OPCs. So essentially the stacked bar graph shows this very nicely. The pinks are the astrocytes and the yellows are the oligodendrocytes. And you see how by removing FGF signaling, you push the cell farther to oligogenesis. So the summary of this part is simply, I introduced the idea of a neuron-glia cell fate switch, but what we uncovered is that while when a cell loses FOXG1, it becomes permanently gliogenic. There's no going back, right? But that loss of FOXG1 makes astrocytes only if FGF signaling is still available. If FGF signaling is also not available, we actually now make a different type of glia, oligodendrocytes. So we've sort of completed the progeny output of this mother cell. So now we have not one or two, but 3 types of mother cells, each changed by the gene regulation in it. So this is the first story of the first gene. And I'm going to switch to LHX2, the other gene, which also connects with gliogenesis in very interesting and completely different ways from FOXG1. Yeah? May I have a sense of time? Am I going too slow? What is 0? I haven't talked for half an hour, 45 minutes. All right. Okay, I have another 15 minutes of thing. Are you all with me? Yeah, I don't mind getting asked clarification questions midway, okay? It's very different, no? I realize medical school talks are very formal. Thank God there's nobody in those judges' seats. Okay. All right, LHX2. We've been working with phenotypes of this transcription factor in the mouse model for a while. And recently, last year, in fact, we reviewed this paper. We realized that there are human studies, and I don't know why this is black. I will change the background. Or actually, I can just change this because you can't read it otherwise. There. And there. Okay, so this study found this, I don't want that, sorry. Okay, this study found human patients with LHX2 haploinsufficiency. Okay, in the mouse we don't see a haploinsufficiency phenotype, so we knock out both genes. But in humans, losing one copy seems to be enough. Okay, they identified 19 individuals from 18 families carrying a small chromosomal deletion, very likely gene disrupting or missense variants of LHX2. In this paper, they analyzed exactly what the problems are, and there's a very detailed, beautiful analysis. But suffice it to say right now that this is relevant because affected individuals present with autism spectrum disorder, variable intellectual disability, and microcephaly. Okay, so there are humans walking around with LHX2 phenotypes, and I'm going to talk about one of the various phenotypes that we have reported in the past years, a gliogenic phenotype. Okay, so here we have the same neuron glia production, and now we're going to focus on this progenitor, postnatal day 1, at which point it is making only glia. This progenitor expresses LHX2, and we're going to examine what happens and what does the LHX2 do in this progenitor. And the answer is that LHX2's job is to control the amount of glial proliferation. Okay, that loss of LHX2 makes the glia overproliferate. This is the work of a DST-inspired faculty fellow working with me, a very talented scientist, Archana Iyer, very ably supported by JRF Rian Frontiero. Okay, and here is postnatal day 1. You can see LHX2 is expressed in the progenitors, also in postmitotic neurons, and in fact in postmitotic glia as well. And here, for example, is ALDH1L1. All the blue cells are glia, and you can see LHX2 is expressed in, I shouldn't say postmitotic glia, but differentiated glia, because astrocytes are not postmitotic, they divide. So LHX2 is at high levels in these astrocytes, and I'm going to show you that its job is to restrain how much they proliferate. Okay, the model we are using is a HEFAP CRE-ERT2, which is inducible by tamoxifen. Whenever we give tamoxifen, the CRE is active in a LHX2 conditional animal, LOX-P sites, and we've crossed it to an AI9 reporter so that all the red cells are those that have lost LHX2. Okay, that's just our readout. Okay, so here we go. Tamoxifen at postnatal day 1, remember this is a P1 progenitor, analyzed 15 days later. In control, you simply see the number of cells that happened to have experienced the CRE and gotten recombined, and they are red. And they're all astrocytes because at this age, the progenitors are making only astrocytes. And now look what happens if simultaneously we delete LHX2. Whoa, carpet of red cells. Okay, rampant gliogenesis, and this is overproliferation. If we look quickly 5 days later, using Ki67, which is a marker for proliferating cells, you can see in the control there is only a little proliferation, but in the mutant, much, much more proliferation. So all of this rampant gliogenesis is due to overproliferation, and it tells us LHX2's job normally is to restrain proliferation. Is this relevant? Well, turns out that we found this paper, Cheng and colleagues, which basically looked at transcription factors present in glioblastoma. And looky here, the glioblastoma signature of transcription factors that go down includes our candidate LHX2. So again, an LHX2 loss model, glioblastoma consistent with overproliferation. So this is one medical connect of this finding. And in the rest of the talk, I'm going to tell you several sort of features of this finding that could be medically relevant. Right now, we're studying them in the mouse model. So first, the mutant astrocytes, LHX2 knockout astrocytes, have shrunken morphologies. Okay, so we began this exploration asking, astrocytes are supposed to be good for neurons. They're supposed to provide necessary support. They're supposed to provide appropriate neurotransmitters. They're supposed to, in fact, even mediate neuron-neuron connection. When two neurons connect with each other, it's not just incoming and an outgoing neuron, it's also an astrocyte, so it's called a tripartite synapse. Okay, astrocytes are critical for neuronal function. Are more of them good? The answer is, if the more of them is produced by loss of LHX2, perhaps not. Okay, so while there are many more astrocytes, their morphologies are shrunken. Here's just a count. And now we're going to talk about multiple effects of how these mutant astrocytes have caused non-autonomous effects on other cells around them or on other features. Okay, so one of them, the same model, HEFAP CRE-ERT2 with AI9 and LHX2 loss. What we also did at this same time is we electroporated at E15. Remember E15, the progenitor is making neurons? We just put a membrane-tagged cast EGFP. So this E15 progenitor was not knockout for LHX2. It was just labeled with GFP because we wanted to label the neurons that arise at E15. And then using the tamoxifen-induced driver, we made the astrocytes mutant later. Okay? And this is what we get when we examine P2. Look at this gorgeous picture, okay? Here is a neuron in all its glory with all its fibers. And look how all these fibers are nicely surrounded by the red astrocytes. They're red because of the AI9. Yeah. So this is really a picture of how astrocytes essentially modulate and sort of support and cover a neuron's arbor. And what we found, and this is the work of Achintya Srivastava, who's a new PhD student, we found that when there are control versus mutant astrocytes around a normal neuron, okay, the neurons are normal, the arbors change, okay? A Scholl analysis of the apical dendrites shows that the mutant dendrites are longer. Or rather, the dendrites of a normal neuron, when it's surrounded by mutant astrocytes, are longer. Fancifully, as if the neuron is trying to seek good astrocytes and not finding them, okay? The basal dendrites, in contrast, show an opposite phenotype. The basal dendrites of a normal neuron surrounded by mutant astrocytes are shorter. So you can clearly see non-autonomous effects of these mutant astrocytes on a normal neuron that's pretty much trying to do its function, but now its dendritic arbor is modulated simply because the astrocytes are not wild type. Okay, this is one finding. Here is another. We examined the blood-brain barrier because astrocytes wrap around the blood vessels, the fine capillaries that enter the brain, and in fact mediate what comes into the neuron space. And here is a very interesting. We simply injected, this is Archana's, she injected Evans blue straight into the heart. And when you inject Evans blue into the heart, the pores will turn blue as evidence that the Evans blue has gone everywhere. Fine capillaries, ears will turn blue, and the brain will turn blue. Does this brain look, well, it looks darkish, right? Okay, the brains turn blue because the fine capillaries have taken up the Evans blue. But look at the mutant. Whatever color you call the control, you can surely see the mutant is a nice pink. Okay, this is telling us there's something wrong with the blood-brain barrier. We do not know what it is. This is completely new data, okay, completely new. Aquaporin 4, okay, a water channel which labels the end feet of the astrocytes on blood vessels. So we're not looking at blood vessels directly. We're looking at astrocytes that wrap around the blood vessels. Okay, here's in the control, and here's the mutant. Okay, we have not quantitated this. This is really fresh data, but you can get a sense that all does not seem to be similar. Okay, there is potentially a phenotype in the mutant that we're trying to map. Is it that the astrocytes don't wrap around the blood vessels properly, and that's why we're potentially seeing more fragmented bits of blood vessels? Or is there a dramatic non-autonomous effect in the blood vessels itself? Because astrocytes are known to, as the endothelial cells come, astrocytes are known to pick one here, pick one there, and join them. Okay, there's live video of this published in the literature. So this fantastic function of astrocytes may be abrogated, or at the very least, the mutant astrocytes aren't wrapping around the blood vessels properly, thereby making the blood-brain barrier potentially tighter. And this would have dramatic consequences. Okay, so this is one more effect of these mutant astrocytes. And finally, what do astrocytes do in seizure? In seizure, neurons undergo excitotoxic damage because of the glutamate they output. Astrocytes are protective. They mop up the glutamate, and they do so using the GLT transporter. The first finding alerted us that all may not be well with, these large numbers of astrocytes could have mopped up the glutamate better, right? So they could have been protective in seizure. But turns out that the GLT1 transporter levels themselves have dropped in the mutant. So having too many astrocytes is not useful if you can't mop up the glutamate. What happens in seizure? Well, what you would predict based on this GLT. Okay, the seizure is scored by this Racine scale. So a drug, this is induced seizure in response to the drug PTZ. Depending on the dose of PTC, okay, you give a normal animal, the animals go through a Racine scale of just jerks or then progressing to clonic-tonic seizure. And the degree of severity is marked on a scale of 1 to 7, where, okay, it begins with trembling and neck jerks, then clonic, and then tonic-clonic seizures, and then finally death. Okay. Archana did a dose response, settled on 50 mg per kg, and here's what happens when control versus mutant astrocytes animals are given seizure. At this particular amount of drug, controls do not go up to Racine scale 4, okay, in this set, but the mutants have progressed, okay. Many of them are in Racine scale 4. And of the set that do progress to Racine scale 5 or 6, again, the mutants are far more in number than the controls. So it clearly seems as if the seizure response, the induced seizure response, is consistent with what we see with the GLT, that animals with mutant astrocytes, LHX2 mutant astrocytes, are unable to protect against seizure compared with controls. And this again is another phenotype with the LHX2 knockout. And the reason I'm so let me just summarize this. Okay, loss of LHX2 leads to overproliferation, and potentially this is relevant in glioma because it was a candidate in glioma. Mutant astrocytes are shrunken, potentially deficient in clearing glutamate, and they have non-autonomous effects on neuronal morphology and function, and possible non-autonomous effects on the blood-brain barrier. So, given that human LHX2 haploinsufficient patients have been identified, and they exhibit a range of disorders from autism to microcephaly, okay, nobody has yet looked at what happens to the astrocytes. If these patients are haploinsufficient for LHX2, potentially their astrocytes may also reveal these deficiencies, and this could explain some of the phenotypes. So that is where this work has a medical connect. Okay, I will close thanking the funding agencies that supported this work. Internal funds from the DAE, Archana's DST Inspire Fellowship, a grant from the DBT, and I'll finally end with, I don't know, an element that sort of conveys our lab's serious but slightly crazy atmosphere. I say this because there are students in here who, you know, might have labs that either resonate or maybe now they will take their labs in this direction. Here are our lab quotes. These are things that people say in their natural environment. They post, for example, "Don't lock the lab, I'm assembling my genome." Or, "There was no signal in my brains." Or they ask, "Are you getting a brain anytime soon?" And "Bro, pluck it like an eyebrow," is one student telling another how to extract choroid plexus from the ventricle of an embryo. And sometimes people are fascinated by the neuron glia cell fate, which. And "Come, I'll teach you sexing" is not a pickup line. We have to discriminate male and female embryos because one of our drivers is on the X chromosome. So teaching sexing is what they do in the Tolle lab, and this is a running joke in the department. You will find these on our website, stollab.co.in. Do take a look and enjoy our science and our fun. Thank you very much.
Tolle, for this very illuminating and fantastic work. I think the journey starts with rare human disease, picks up the genes, and then you go on into a wonderful journey into how they actually mediate neurodevelopment, and then come back to clinical implications. So I think, you know, speaks largely about the oneness of biology, and we are one animal in this whole universe of biology, animal or plant rather, in that sense. So thank you very much for this really wonderful piece of work where you illustrated so many things, so many technologies which would also be very useful to our students. And with this, there's already one person very enthusiastic over there.
Hi, Shubha. It's a very nice talk. So I have one question, that is, duplication of FOXG1 in 14q12, if I'm not wrong, that leads to developmental epilepsy, mental retardedness, or cognitive deficits. So, what I'm asking, like, have you seen any kind of like these kind of experiments? Have you done like deletion of that FOXG1? Later on, you just revert it by tamoxifen-induced re-expression to those cells, mother cells, whether you actually kind of revert them. So I don't know, like these are the patient data I'm saying about these. There are around like rare, like 6 or 7 patients data.
Oh, very cool question. I get what you're saying. Yeah. Did people understand? The idea is to give back FOXG1.
Yeah.
So it's a cool question. To do this in the lab, as you know, you need actually the mouse reagent to do it with. Karina Hanashima's lab has developed a TET-on, TET-off system where she can actually do this. Okay, her current work that's published is in the embryo to take it on and give it back, but that would be the reagent to do this with. We don't have it, so I don't have the answer. But a lot of it is building the tools to ask the question, then you get to ask the question, and this is a very cool one. Thank you.
Shubha, thank you very much. I just wanted to ask you, most of the children would have a short life. You know, they die very young. Have you come across families or familial cases with a history of GBM perhaps in the family, or autism spectral disorder in the family? Because it's a bivalent gene you're talking about. And you're talking about that could be carriers, the parents could be carriers, but this is a new mutations that can arise. The question is how? I mean, how do these new mutations arise?
So the bivalently marked astrocytic genes were FOXG1. FOXG1 syndrome can cause severe autism and many other problems. The corpus callosum is thin or not fully formed, so it's very, very severe. The GBM Connect is the LHX2 story, and FOXG1 syndrome, GBM has not been reported. Because it causes neuronuclear cell fate switch, but not necessarily overproliferation. So there are two separate ones. Depending on the nature of the autism and all of the other peripheral effects, survival can be very short, but it can also be long. So one of the major healthcare concerns is actually how to support a late-age autistic patient. You know, so there's whole sets of healthcare concerns, and actually you're more expert than me.
Thank you for a wonderful talk. And I have a query, like there are many neurodegenerative disorders where neurons are not functional. So is there a mechanism where, you know, astroglial cells can be converted to neurons and they can be used for, you know, kind of, especially for the treatment of such diseases? So where we don't have any options left in many of the neurodegenerative disorders.
I mean, thank you. That's actually the question at the cutting edge of technology. Reprogramming of cells.
Yes, exactly. That's what I meant.
So, I mean, people now don't need to take astrocytes and make them back to neurons, because induced pluripotent stem cells can be programmed to produce neurons, and that's a whole area of expertise. So making the neuron is one thing. Planting it into a brain and expecting it to develop the circuitry is another. So that is, to my mind, the challenge, because you can replace, for example, interneurons which have more local connections, but how do you replace something that has a long-range connection? Okay. You know, so the challenges are that of integrating into circuitry. Okay. Right? I mean, there are in vivo models. As you know, the nasal epithelium, olfactory sensory neurons, regenerate as a matter of course. They're exposed to the environment, they die, they regenerate, and those axons find their way back through the cribriform plate of the skull to the olfactory bulb as a matter of course. Okay, so, I mean, robust guidance mechanisms must be in place, because normally cells are not expected to find, so circuitry is the problem, and I don't have the answer.
Okay, so, thank you.
So young folks, that's where you have to figure things out.
Thank you very much.
There's a question here, sorry.
This is, I'm a physiologist, so I'm wondering who starts functioning first, neuron or astroglia? According to scale, they will come up later in large numbers. So I think housekeeping and scaffolding and mopping potassium or glutamate, whatever, is more important than functioning. So how come this reversal takes place? Either by the growth of the neurons at later date, astroglia, they come, they supersede in speed. So I was wondering why the reverse scheme by nature.
You think it would have been more logical for the astrocytes to be formed first?
Yes, obviously,
I
housekeeping.
mean, you know, as is well said, in biology we don't ask why. but the neurons are electrically active, and early electric activity is in fact important to set up the circuit. So for example, in the rodent model, the somatosensory barrels come up in the early postnatal life. And so there is electrical activity, but the circuit cannot be fully functional until it's myelinated. The signal will not go very far. So, I mean, I guess this is why gestation lasts as long as possible, to make the brain reasonably okay before its output. And even then, for humans, as you know, the baby's output when the skull is just big enough that it won't kill the mom. But the brain is seriously underdeveloped, which is why our babies just lie there looking awfully cute. That's their survival mechanism. But the brain is actively wiring itself. Cells are still migrating. Myelination is still happening massively in the human baby. And only then does the brain actually begin to do things. I mean, we see it every day in our homes.
I've had a lot of hand gestures from Dr. Bal. So last crisp question, and please, I am getting pressure
But
backstage.
Dr. Bal, short focused question and short answer.
Ma'am, it is very, very interesting that you have challenged the whatever we taught in the medical schools. We have told that the stem cells is around the ventricles, and they first create the scaffold on which the neurons migrate periventricular to. That's why the heterotopia and all these disease being described in neurology, which is a standard teaching. But your hypothesis is challenging the foundation of neurodevelopment.
No, I haven't challenged anything.
No, you are telling first it is in E15.5, means in the 15th day of embryogenesis, the neurons first, and then it translates to the glial cells in the latter part of the day. But it is taught to the medical school students reverse way.
No.
You ask the neurologist, they can answer.
No, the stem cell, that is the E15 progenitor, is otherwise known as a radial glial cell. The word glia there is a misnomer. People thought they were glia. A radial glial cell is like so. And when one becomes 2, one continues to be a radial glia, and its postmitotic neuron migrates using the glia as a scaffold. So all is well. All is well. You don't have to throw out your textbooks.
Wonderful discussion, and you aroused a lot of interest and a lot of thinking in the audience. With this, I think.
So 1 second. Yeah. Thank you very much, ma'am, for an interesting talk. I now request the chairpersons to kindly felicitate Dr. Shobha Tole, ma'am. Thank you, Professor Mehra and Professor Subrata Sinha, sir, for kindly chairing the session. I now hand over the stage to my colleague, Dr. Yogita, for the next session. Thank you.
Thank you, Dr. Kamal. The next segment is Innovative Research, Best Papers by Academic Junior Residents, MD, MS, and MDS students. It's an honor for inviting for this chairing this session Dr. K.K. Deepak and Dr. Shinjini Bhatnagar. Dr. K.K. Deepak is former Dean Examinations and Head of Department of Physiology, AIIMS, Delhi. Welcome, sir. Dr. Shinjini Bhatnagar is former pediatric gastroenterologist and a renowned Indian scientist, AIIMS, Delhi. She is currently Distinguished Professor, Maternal and Child Health at THSTI. Welcome, ma'am. I request the chairpersons to now kindly conduct the session.
Good morning, and we begin the session. So this is Ignited Minds, and we're very happy to have that MD students, MS and MDs, they are presenting. So the first rules of game, we're going to stick to the timings, and we'll take quick questions. And so we begin with the first presentation, and this is by Sana Fazil, Department of Biochemistry. Yeah, so you have 7 minutes time, and please come on the dais and give your talk. Thank you.
Very good morning to all of you. So before I start, I would like to thank the Institute for giving me this wonderful opportunity. So I'll be talking about premyelinating oligodendrocytes exposed to hypoxia lead to a maturation arrest in a cell line model. So cerebral palsy is a permanent, non-progressive neuromotor disorder of cerebral origin that is characterized by abnormal tone, posture, and movement. So it is a hypoxic ischemic injury to the brain that is a major cause of cerebral palsy. And it's been found that the incidence of cerebral palsy increases with a decrease in the gestational age. That is, the most common risk factor for cerebral palsy is the prematurity. So during 24 to 32 weeks of gestation, it is a premyelinating oligodendrocyte that are predominant in the developing brain. So these are the progenitors that generate mature oligodendrocyte that is responsible for the production of myelin sheath in the central nervous system. So these are the stages of maturation of oligodendrocyte. And among these 4 different stages, it is the premyelinating oligodendrocyte that is the most vulnerable to hypoxic injury. And so the window period is identified as 24 to 32 weeks of gestation. And each stage will have its own characteristic marker. So NG2 is the characteristic marker of premyelinating oligodendrocyte, and MBP is a characteristic marker for mature oligodendrocyte. So for my study, I've used a cell line which is called MO3.13 that resembles premyelinating oligodendrocyte, and that can be differentiated into a mature oligodendrocyte by using a chemical called PMA over a period of 7 days. So aim of the study was to investigate the impact of hypoxic injury on the survival and differentiation of premyelinating oligodendrocyte using a cell line model, using a cell line resembling premyelinating oligodendrocyte. So we have created a disease model for cerebral palsy by exposing undifferentiated MO3.13 to hypoxia for 48 hours, and the controls were exposed to normoxia for 48 hours. Then we have validated hypoxia by measuring the expression of hypoxia responsive genes like CA9, VEGF, and PGK1 by qPCR. So these are the downstream effectors of HIF signaling pathway, that is hypoxia inducible factor, that in turn is activated by hypoxia. So we found that at an oxygen concentration of 0.2%, the expression of CA9, VEGF, and PGK1 has been increased compared to those cells exposed to normoxia. So this helped us to confirm the exposure of undifferentiated MO3.13 to hypoxia. Then we have also evaluated apoptosis in undifferentiated MO3.13 exposed to hypoxia and the controls exposed to normoxia for 48 hours. And we have stained the cells with annexin 5-HC and propydium iodide dye. So during apoptosis, there will be flipping of the phosphatidylserine, which is normally present in the inner leaflet of the cell membrane. So this externalization of the phosphatidylserine, so the annexin 5 dye will go and bind to this particular phospholipid. Then propydium iodide dye was used to exclude non-viable cells. So this is our result. We have selected the population of cells. Then we have selected the singlet cells, followed by gating. So those viable cells are positive, negative for both the dyes. Then early apoptotic cells were stained only for FITC, and late apoptotic cells were stained for both the dyes, while necrotic cells was positive only for propydium iodide. So we found that 26.9% of cells underwent apoptosis in hypoxia exposed cells, compared to those exposed to normoxia, which was only 7.21%. So this shows that when premyelinating oligodendrocytes are exposed to hypoxia, it can lead to apoptosis of those cells. Then we have analyzed the effect of hypoxia on differentiation of prioles into mature oligodendrocytes. For that, we have exposed undifferentiated MO3.13 to hypoxia and controls exposed to normoxia for a period of 48 hours. And then we have differentiated using a chemical called PMA for over a period of 7 days. Then we have captured the morphological images with the differentiation at different time points like day 0, 3, 5, and 7. And initially, we observed stellate-shaped cells, and with the differentiation, we have observed development of long and thick processes. Then those exposed, then undifferentiated MO3.13 exposed to hypoxia, we found that the cells were highly distorted after exposure to hypoxia, and with the differentiation, the development of process was found to be hampered. Then using qPCR, we have analyzed the expression of NG2 and MBP, which are the characteristic markers of preoles and mature oils respectively. So we found that the expression of NG2 was found to significantly decrease with the differentiation, and there's an increase in the expression of MBP significantly from day 0 to day 7. So this shows that at normoxia, the premyelinating oligodendrocyte will differentiate into a mature oligodendrocyte. But after exposure to hypoxia, it's been found that the NG2 expression did not decrease significantly compared to those NG2 expression at normoxia, and the MBP expression, instead of an increase, we have observed a decrease in the expression in those exposed in undifferentiated MO3.13 exposed to hypoxia. So this confirmed that when the premyelinating oligodendrocytes are exposed to hypoxia, it will undergo a maturation arrest. So this is the conclusion of the study. So premyelinating oligodendrocyte, when they are exposed to hypoxia, they will undergo maturation arrest as well as apoptosis, and this will lead to a failure of myelination that can lead to development of cerebral palsy in later life. So the translational value of this study is therapies that can promote the survival and differentiation of preoles after hypoxic injury that can be beneficial in those disorders which involve hypoxic injury like cerebral palsy. So I would also like to thank DBT for the funding, as well as my guide, Dr. Sudeep Sen, Professor, Department of Biochemistry, and my seniors for their constant support. Thank you.
Thank you, Sana, for this. So we'll take up one question, quick question from, yeah. So youngsters from the back. Or anybody from the front. So just my question, why it is a contrary result, like hypoxia benefits?
Sorry, sir?
How the hypoxia is beneficial?
How hypoxia is beneficial? So
people take hypoxia to stimulate cells.
So in our study, we found that hypoxia is leading to hypoxic injury that is preventing the maturation of premyelinating oligodendrocyte to a mature oligodendrocyte. So in our case, this is not beneficial for our cells. So this is making the developing brain vulnerable to hypoxic white matter injury, and this can further lead to the development of cerebral palsy in the later period of time.
Okay, thank you. Thank you.
Thank you, sir.
We will go over to the next one.
So I'd like to invite Dr. Hannah Karim, Department of Pediatrics. She's going to talk to us on development and internal validation of a primary caregiver reported instrument for neurobehavioral assessment of children with epileptic encephalopathy. Over to you, Hannah.
Okay, good morning all. My topic is development of a questionnaire-based tool to measure the primary caregiver reported outcome in children with epileptic encephalopathy. The epileptic encephalopathies are disorders associated with significant cognitive and behavioral issues, and there is no such patient-reported outcome measures that address these domains, and it is important that these need to be studied for evaluating the efficacy of any intervention and for the prognosticating and assessing the effect of any intervention on a short-term basis. That forms the background of my study. So aim was to develop a questionnaire-based tool with a good internal consistency, test, retest reliability, and construct validity to assess the outcome in children with outcome in terms of improvement in cognition, behavior, and social personal skill in children with 4 to 14 years with drug refractory epilepsy and epileptic encephalopathy. And the secondary objective was to study the correlation between the expert clinical impression of severity of neurobehavioral and social personal skill impairment on a 5-point Likert scale, along with the correlation between the spike wave index in EEG and the available other scales like Gaze scale, VSMS, along with the newly developed patient-reported questionnaire. Inclusion criteria included age group of 4 to 14 years with a social quotient less than 70 with established electroclinical diagnosis of one of the 3 conditions, Lennox-Gastaut syndrome, ESES, and drug refractory epilepsy according to the definition of International League Against Epilepsy 2022 definitions. So the exclusion criteria included a child diagnosed with a progressive neurological illness or any child suffering from chronic illness that can affect the cognition, behavioral, social, personal skill, or child had a convulsive status epilepticus in the last 1 week, or any illness leading to the hospitalization of any duration in the last 1 month, or the primary caregiver not staying with the patient over the last 1 month. So sample size was calculated according to the standard formulas for kappa agreement, factor analysis, and Cronbach alpha. And I have enrolled 101 patient. 101 patient because of the time-bound effect of the MD thesis. Coming to the tool development, for content validation, extensive literature review was done regarding the cognitive, behavioral, and social issues in children with epileptic encephalopathy. And along with that, a reviewing of the existing questionnaires. And I have interviewed 10 children with epileptic encephalopathy on issues faced and expected outcome related with the therapy. In that, a pool of questions were collected, and it was discussed in-house in the pediatric neurology division, after which the version 2 of the questionnaire was developed. It was circulated through modified Delphi method among review committee including the following experts, and modification was done in the form of revision of the sentences, and a timeline for responses were also added. And after modification, it was circulated for approval, and it was translated to Hindi and back translated to English for the bilingual validation. For phase validation, this questionnaire was run in 10 patients as a pilot run, and further modification was done in the form of simplification of words. And this was again circulated for the approval, and it was translated to Hindi and back translated to English. And this becomes the final questionnaire, which contained 20 questions with a minimum score of 20 and a maximum score of 140. And this questionnaire was run in field, as I mentioned, it was run in 3 study groups, LGS, ESES, and drug refractory epilepsy at 2 time points. So coming to the demographic characteristics, I have enrolled patients from 4 to 14 years of age with a median of 8, median age of 8 years, and among that 62% had a structural etiology, and 71% was having ongoing seizure at the enrollment. Coming to the questionnaire properties, Cronbach alpha for the 20 question was 0.9, which was showing an excellent internal consistency, and the inter-rater kappa agreement between the responses to the questions at baseline and at 7 days for whole question, which was showing substantial to perfect agreement among the all 20 questions. After which the factor analysis was done. Based on the eigenvalue and the scree plot, we were able to classify these questionnaires into 2 groups or 2 factors, and the factor loading was done for these 2 groups, and we can see that after factor loading, these 20 questions can be classified into 2 groups. Question number 11 to 14 was classified in 1 group, and the rest of the question was classified to the other group. And while seeing retrospectively into the questionnaire, the question number 11 to 14 was seeing the behavioral issues associated with these children, and the rest of the questions was showing the cognitive, social, personal skills, social, personal issues associated with the epileptic encephalopathy patients. Coming to the secondary objectives, the correlation of the total score of the questionnaire with expert opinion was done at the baseline and at 3 months. It was showing a significant correlation with a correlation coefficient of 0.7 at the baseline and 0.8 at the follow-up. And coming to the correlation of the developed questionnaire with the GAY score, SWI in EEG and VSMS score, it was showing that the correlation was significant for the GAY score and VSMS, but there were no correlation found along with the SWI in EEG, both at the baseline and the follow-up. So key highlights of the questionnaire is validation and reliability of the questionnaire demonstrate a good internal validity, test-retest reliability in assessing the cognition, behavior and social personal skill in children with epilepsy from 4 to 14 years. And coming to the construction methodology, it included meticulous process involving the patient input, literature review and the expert consultation, and there is a strong internal consistency, and there is and a distinct factor loading was noticed, and the questionnaire can be grouped into two broad domains, including cognition and social personal skill as one group, and the second group as behavioral dimensions. And this shows and along with that there is a correlation with established scales like a GAY scale and VSMS, and the validity was supported by correlation with the expert opinion. And still the external validation on a large participant group is needed for the clinical and research recommendation for this questionnaire. Thank you. Along with that, I would like to thank my guide he was supporting from beginning to towards the end of whole the journey. So I would like to thank my guide, Dr. Prashant sir, along with the whole Department of Pediatrics. Thank you.
Thank you very much. We have time for a couple of quick questions. No questions. We'll be asking them ourselves then. Okay, Hannah, that was very interesting. Can you hear me? Yeah.
Yes.
So you said your sample size was not what you wanted it to be.
Yes.
So can you tell us the problems with that? I mean, eventually in your analysis, interpretation?
In interpretation, we were able to, for factor analysis, we can see the KMO value was reaching up to 0.9, which was saying that the sample size was adequate, even though we are not able to reach the expected sample size of 296.
Okay, and if there are no more questions, I'll just have one more. There is somebody there? Yeah.
Drug-resistant epilepsy group, you had just 16 or 17
Yes, ma'am.
children, and the others were relatively more.
It was because, ma'am, in drug refractory epilepsy, I have included the patient with the social quotient of less than 70. In the study population, it is for LGS and drug refractory epilepsy. This patient was this group of patients are having significant involvement of the cognitive and behavioral issues. But in drug refractory epilepsy, only small chunk of population. Even though there are lots of drug refractory epilepsy patients, the patients who is having significant cognitive and behavioral issues are seen in only in small chunk of patients. That's why the sample size was less.
I was even wondering why you'd include them in this study. They are very different from the other participants of your, but they are in themselves very heterogeneous.
Yes, ma'am, but still we have noticed that there are significant issues with the cognition and behavior in this group of patients also, ma'am, even though the number are less, ma'am.
So we'll go on to the next one. Okay, thank you very much, Anna. We can give her a big round of applause.
So the next speaker is Dr. Lanu Mona Aimal from Department of Biochemistry. Please go ahead.
Good morning, everyone. My work is on the epigenetic regulation of FAT1 expression via DNA methylation in glioma. So our lab for the first time identified the oncogenic role of FAT1 in glioma. It promotes immunosuppressive tumor microenvironment, increases migration and invasion and EMT stemness, and causes activation of pro-inflammatory molecules by suppressing PD-CD4 and PD-CD10. So DNA methylation is a post-replication modification occurring on the cytosines of CpG dinucleotides in the promoter region, which results in repression of gene expression. So hypermethylation of tumor suppressor gene can cause its silencing and lead to cancer. Also, hypomethylation of proto-oncogene can activate oncogene and cause cancer. DNA methylation is done by DNMT enzymes, mainly DNMT1, 3A, and 3B. Demethylation is done by the TET enzymes, TET1, 2, and 3. These are the list of some cancers with overexpressed DNMTs and reduced TET expression, including glioma. But till now, there is no report on the expression correlation of DNMTs and TETs with FAT1 expression and the methylation status of FAT1 promoter in any cancer. Therefore, we have analyzed the promoter methylation status and correlated it with the expression of FAT1, DNMTs, and TETs in cancer cells. So this was my work plan. First of all, we analyzed the mRNA expression of FAT1, DNMTs, and TETs using qPCR and their correlation in both cancer cells and glioblastoma samples, followed by analysis of FAT1 promoter methylation status. Then we looked for the effect of DNMT inhibitor on FAT1 expression. So first, we analyzed the expression of FAT1 in different cancer cell lines of glioma, pancreatic cancer, cervical cancer, and hepatocellular carcinoma. And we found high expression of FAT1 in all the cancer cell lines except for T98, U87, and MIAPACA. This was followed by expression of DNMTs in these glioma cell lines, and in all the cell lines, there was high expression of DNMT1 and 3B and low expression of DNMT3A. As for the TET enzymes, TET2 was low in all the cell lines, and high expression of TET1 and 3 was seen in these cell lines, except for 2, which showed low expression of TET1. Similarly, the expression of FAT1, DNMTs, and TETs were also done in glioblastoma tumor samples, and among the 7 samples, all of them showed high expression except for 1 sample, and all the 3 enzymes were high in most of the samples except for 2 samples which showed high expression of DNMT3B. And for the TET enzymes, all the 3 TET enzymes were high in all the samples except for 1 which showed very low TET1 expression. Now, expression of DNMTs and TETs were also done in 2 pancreatic cancer cell lines, one with low FAT1 expression and another with high FAT1 expression. And all the 3 DNMTs and TET1 was high in both these cancer cells. Now, using this qPCR data, we did the expression correlation of FAT1 with DNMTs and TETs in glioma cell lines, and we found a trend of negative correlation between FAT1 and DNMT3B and a positive correlation between FAT1 and the 3 TET enzymes, meaning high FAT1 expression was associated with low DNMT3B and high TET1, 2, and 3. These similar findings was also seen in case of the glioblastoma tumor samples. So this suggests a possible role of DNMT3B and the 3 TET enzymes in regulation of FAT1 promoter methylation. Next, now we analyze the FAT1 promoter for looking at the methylation status. Using embossed CPG plot, we first identified 3 CPG islands in the promoter region, and for our study, we have selected island 1 lying closest to the transcription start site and island 3. This was followed by designing bisulfite-specific primer using the MITH primer software for the 2 islands. And bisulfite conversion and sequencing was done as follows. First, DNA isolation was done, followed by bisulfite conversion. So when DNA is treated with sodium bisulfite, the unmethylated cytosine is converted to U, which then on PCR is converted to C, while the methylated cytosines remains as C. So this was followed by PCR amplification and sequencing. This is a representative chromatogram of the CPG1 island, and out of the 9 CG sites identified in the region analyzed, all the Cs were converted to Ts in the 3 samples, meaning all of them were unmethylated. So CPG1 showed no methylation in samples with both high and low FAT1 expression, but in case of CPG3, those samples and cell lines with low FAT1 expression showed high methylation status, and those with high FAT1 expression showed low methylation status. So this suggests that CPG island 3 methylation play a role in the regulation of FAT1 expression. Now then we looked for the effect of 5-azad-2-deoxycytidine, which is a DNMT inhibitor on FAT1 expression using 2 glioma cell lines, one with low FAT1 expression and one with high FAT1 expression. And after treatment, we found increased expression of FAT1 with LN229 requiring higher dose of the drug. So this reflects the role of promoter methylation by DNMT in regulating FAT1 expression. Now, on the basis of our observation of a negative expression correlation of FAT1 with DNMT3B, positive expression correlation with the 3 TET enzymes, the promoter hypermethylation in CpG3 of low FAT1 expressing GBM tumors or cell lines and vice versa, and also the increased FAT1 expression on DNMT inhibition in glioma cell lines all suggest a role of FAT1 promoter methylation in regulating its mRNA expression. I would like to acknowledge my supervisor, co-supervisors, and my lab members. Thank you all.
This is open for one question from the audience. Anybody has a question? And if there's no question, we conclude this session. We can clap again for nice presentation. Thank you. Over to Dr. Sanjini.
We thank the chair and the speakers and the judges for conducting an interesting session. We conclude this session, and I now hand over to Dr. Sanjukta for announcing the next segment.
We welcome you to this session where now there will be platform presentations by senior residents. It is our honor and privilege to have Dr. Rama Chaudhary and Dr. Meenu Bajpai as the chairpersons for this session. Professor Rama Chaudhary was former Dean Research as well as head of the Department of Microbiology at AIIMS New Delhi. She is currently working as ICMR Emeritus Scientist in the Department of Microbiology, AIIMS New Delhi. She is also the American Society of Microbiology's International Ambassador to India and has numerous publications and awards to her credit. Welcome, ma'am. Professor Meenu Bajpai is currently the Executive Director of the National Board of Examinations in Medical Sciences. He formerly held the position of Dean Academics and Head of Pediatric Surgery at AIIMS New Delhi. He has received several awards including the Kamla Menon Medical Research National Award of ICMR and the Ramanna Fellowship of the Ministry of Science and Technology. He has several research papers and patents to his credit. Welcome, sir. I now request the chairpersons to please conduct the session.
Hello. So good morning, everyone. We are starting this session, Ignited Minds Best Papers by Senior Residents. And the first presentation is by Dr. Akhil Santosh, who is from Medical Oncology. And his topic is randomized double-blind placebo-controlled study of topical diclofenac in prevention of hand-foot syndrome in patients receiving capecitabine, and this is a DTORCH study.
Good morning, everyone. I'm Dr. Akhil, Senior Resident from the Department of Medical Oncology. I'll be presenting the results of the DTORCH study, the randomized double-blind placebo-controlled study of topical diclofenac for prevention of hand-foot syndrome in patients receiving capecitabine chemotherapy. So a little bit about the background. So as you all know, capecitabine is an extensively used oral chemotherapy drug, mainly in the gastrointestinal and breast cancers. And hand-foot syndrome is a very common side effect associated with capecitabine drug. And it is also known as palmoplantar erythroderma. And the incidence of which is around 50%. And it is a very painful and distressing condition. It is not life-threatening per se, but is associated with excessive morbidities because it leads to frequent dose reductions and discontinuations of the chemotherapy drug. So you can see here, this is a classical hand-foot syndrome. It is characterized by extensive edema, erythema, and ulcerations of both the hands and the feet. So till date, there are very limited effective pharmacological interventions for prevention of the same. The only drug that has been found to be effective in an RCT was celecoxib. So that, as you all know, is an oral NSAID, it's a COX-2 inhibitor, but it is not frequently used in the clinics because of its severe gastrointestinal and cardiac side effects. And as you all know, majority of the cancer patients, they are elderly and they will not tolerate a prolonged course of the same. So so the mechanism for hand-foot syndrome is basically because of COX-2 level expression, overexpression. So as you all know, that is why celecoxib was found to be effective. So that was the basis for our hypothesis. So as you all know, diclofenac is also a COX inhibitor. So we formulated that a topical preparation of the same would be helpful in prevention of the same side effect. So this was a study schema. So DTOT stands for diclofenac topical in reduction of capecitabine-associated HFS. So we included patients of more than 18 years of good performance status with breast and GI cancers, and we randomized them 1:1 to receive either 1% topical diclofenac gel or the placebo. And the primary endpoint was incidence of grade 2 or more hand-foot syndrome with several secondary objectives, which I'll be listing. So the secondary objectives was to compare the effect of topical diclofenac with the placebo for preventing any grade of HFS, grade 1 to 4, the time to develop HFS from the start of the drug, the dose reductions of the chemotherapy, the adherence with application, and the quality of life and with the patient-reported outcomes with the HFS 14 questionnaire. So all the participants, they were instructed to apply either 1% topical diclofenac or the placebo, and the dose was 1 fingertip unit, that is 0.5 gram on both sides of the hands. And we did not apply on the foot because the literature says that the incidence is more common in the hands compared to the feet. And so the study duration was for 12 weeks of the 4 cycles. 1 cycle is 3 weeks, or until the development of grade 2 or more hand-foot syndrome. So this was the schedule of assessments. So we initially screened the patients at the baseline. We explained the study to the patients and took an informed consent. And at every visit, we would assess the patients for the side effects, mainly the hand-foot syndrome. We would take the clinical photographs of all the patients. And the other side effects like any mucositis, diarrhea, myelosuppression, they were also assessed. And any dose reductions of the chemotherapy were noted. And the quality of life assessment was done using questionnaires and specific HFS 14 questionnaire, and the adherence to application of the topical drug was also noted. So the assumption for the sample size was that the normal incidence of capecitabine-related hand-foot syndrome is around 30% in the Indian population. So we hypothesized that our study intervention would reduce it to around 15%. So with a power of 80% with a two-sided alpha of 0.05, we included around 120 patients, and accounting for a 10% iteration, the total sample size was 264. So initially we assessed 311 patients for eligibility, out of which 264 were randomized 1:1 to receive either topical diclofenac or the placebo. So coming to the baseline characteristics, so the results, and the median age for population was 47 years. Around 70% were females because we had a lot of breast cancer patients, and majority of them were of good performance status, and around 40% received only capecitabine chemotherapy, and around 60% received combination. And around 56% were breast cancer patients and 43% were GI patients. And majority were stage 3 and 4 because this is the stage where the drug is indicated basically. So coming to the primary results, so here you can see that the incidence of grade 2 or more hand-foot syndrome, it was only around 3.8% in the diclofenac arm compared to around 15% in the placebo arm. The results are significant with a P of around 0.003. So that means there was a 75% reduction in the incidence of developing grade 2 or 3 hand-foot syndrome. So coming to the secondary outcomes, the incidence of any grade HFS, that was also significantly low in the diclofenac arm, around 6.1% compared to around 18% in the placebo arm. And here you can see this important slide that the dose reductions of the chemotherapy, they were very less in the diclofenac arm compared to the placebo arm, around 3% versus 13%. And the adherence to topical medication was similar in both the arms. There was no difference. And coming to the quality of life, so we developed the HFS 14 questionnaire. So here you can see that the score, the QOL score was higher. It was 8.7 in the placebo arm compared to 2.3 in the diclofenac arm. That means a higher score indicates a worse QOL. So the QOL was worse in the patients receiving placebo compared to diclofenac. Regarding the other side effects like diarrhea, mucositis, myelosuppression, they were not significantly different in both the arms. They were similar. So concluding my presentation, we found that such a simple, cost-effective, and a novel intervention like a topical diclofenac gel, which is actually the Voveron gel that you get in the outpatient departments. So it significantly reduced the incidence of grade 2 or 3 hand-foot syndrome. And all the secondary outcomes aligned with the primary. There were lower incidences of all grades of HFS with lesser dose reductions of chemotherapy with a better quality of life. And the benefit was consistent across all the subgroups. Yeah, so we had presented the primary results in last year's American Society of Clinical Oncology annual meeting 2023. It received the Best Oral Abstract Merit Awards. And all the subgroup analysis, they were presented in the European Society of Medical Oncology Congress, and we have received a merit award for the same. Our intervention, it has already come into the guidelines, both in the Medscape and the UpToDate, you can see the same. It was hailed as the most practice-changing study at ASCO 23, and is currently the new standard of care. So I would like to thank all my patients who had consented for the study, my chief guide, Dr. Atul Batra, additional professor, Department of Medical Oncology, and all my colleagues and senior residents who had helped me in recruiting all my patients. Thank you.
So thank you for nicely presenting and concluding in time. So any questions from the audience or any queries? How many times you were applying it topically?
Daily, ma'am.
Daily, how many times?
Twice a day.
Twice a day.
Yeah, so the drug schedule is like, it's 1 gram per meter square. You give it for 2 weeks out of 3, 2 out of 3 weeks. So you give a 1 week rest period. So the patients have to apply twice a day for 14 days. Yeah.
Okay.
Dr. Rajiv?
Thank you for your study.
Yes, please.
Was there any blinding at any site for the primary endpoints?
Yes, it was blinded. The study
So
was
who all
blinded.
were
So
blinded?
both the patients and the primary, I was blinded. Both the primary investigator
And the
was blinded.
packaging was
Yes.
equivalent?
It
for
was a
both?
sealed envelope. So we had sought help of AIIMS laboratory. So they had helped in the blinding and the concealment, and they had device decoded. So it was very
No. The packaging for the drug was
yes
was similar.
Both the size, consistency, everything was same for the diclofenac
thanks.
tube and the placebo, so that nobody could make out any difference.
Perfect. Thanks.
Good.
Yes, yes.
Okay, so again, I think it's about, so you mentioned
Please speak loudly.
you mentioned that you used only a particular brand of the preparation.
Yeah.
So were there any additives? You know, many of these preparations have a few other additives because, you know, they're for relieving pain, and there would be a few other additives. So a comment on that. And I think I'm not still very clear, means the question, so you blinded the, you know, let's say you had similar appearing formulations.
Yes.
Another question would be that, you know, when you apply something on hand, what was the period of contact? How long did you ask them to keep it on? It's not a great preparation to keep on your hands for too long.
We asked them to apply for at least 2 hours and then to wash it off, both in the morning and in the evening. And we had used the diclofenac gel prepared by the AIIMS laboratory, so that came in the brand name of Enzoflame. So basically, whatever preparation you come across in the market.
Not Voveran, right?
Not Voveran, yeah. So I just said Voveran because I think many of us will be familiar with that. But the additives that are used are similar in almost all the brands. Basically, it has menthol, benzyl salicylic acid. The contents are almost the same,
And
apart from the diclofenac.
And your placebo had these contents?
Placebo, yes. The additives were the same in both the placebo and the commit drug. So patients could not make out.
And
So
one of the assumptions was that your, you know, the rates in the control arm are going to be 30%.
Yes.
But you actually got only 15%.
Yeah.
So any thoughts on that?
Yeah. So there were a few studies because, as you all must be knowing, the incidence of hand-foot syndrome is more common in the Asian population. So there were a few studies from China and Japan where they seen that, like educating. So whenever I recruited a patient, so I would just give them a 10-minute educational session just on hand-foot syndrome. So how to prevent it, what is it, and what can you do with use of
be brief,
moisturizers,
please.
avoiding.
Be brief.
So it has been seen that just giving educational sessions to patients before you start the capecitabine, it per se leads to reduction in hand-foot syndrome. So we did not get the expected 30-40% that was actually reported in literature. We had a lesser percentage.
That was more efficacious, possibly.
Yeah, Dr. Lodha, just a corollary to Dr. Lodha's question, were there any non-responders, and did you see any tachyphylaxis in patients? Because these patients require it often, and local applications have this kind of...
We did not see any tachyphylaxis or any other side effects associated with topical diclofenac.
Right, okay, thank you.
Thank
And
you.
we move on to the next talk.
Yes.
Thank you.
This is again a good talk. So the use of sequencing actually in clinical application in substratifying arthrogryposis multiplex is by Dr. Monica Indra Kanti from the Department of Pediatrics, and the title is like this: Delineation of phenotypic and genotypic spectrum of a large Indian cohort of arthrogryposis multiplex congenita using next generation sequencing.
Thank
Please.
you, sir. Good morning, respected dignitaries, teachers, and my dear colleagues. So the title of my study is Delineating the Phenotypic and Genotypic Spectrum of Arthrogryposis Multiplex Congenita using Next Generation Sequencing. My chief guide was Dr. Gupta. So the arthrogryposis multiplex congenita is a non-progressive disorder characterized by congenital contractures involving 2 or more joints in 2 or more different body areas. Various causes are implicated in AIIMS. It can be intrinsic fetal or genetic causes, environmental or maternal causes. There is high phenotypic and genotypic heterogeneity, and about 402 genes have been described in association with AIIMS so far. So, coming to the rationale, very few previous studies describe the utility of NGS in evaluating the genetics of arthrogryposis, and all of them were variable inclusion criteria, and there is paucity of Indian studies. Hence, further characterization of Indian patients with AIIMS is expected to expand the existing knowledge on the condition. So the primary objective was to study the clinical and molecular spectrum of arthrogryposis multiplex congenita using NGS, and the secondary objectives were to study the genotype-phenotype correlation and the functional outcome of novel variants. So this was a descriptive observational study performed in the Division of Genetics, Department of Pediatrics. Patients of all age groups, including fetuses, who fulfill the clinical definition of arthrogryposis were included. Those with amyoplasia congenita and arthrogryposis secondary to uterine structural abnormalities and multiple gestation were excluded. So after patient screening and enrollment, clinical evaluation was done, and clinically, based on the distribution of contractures, they were categorized as distal arthrogryposis, those with predominant distal contractures, pterygium syndromes, those with contractures in addition to pterygium, fetal echinacea, deformation sequence, and neuromuscular causes categorized as FADS NM, and the remaining were categorized as syndromic arthrogryposis. So after sample collection and DNA extraction, probe and whole exome sequencing was performed in all of them. So coming to the whole exome sequencing workflow, following DNA isolation and exome capture and enrichment, whole exome sequencing was performed. The initial data processing was followed by variant calling, variant annotation, and variant filtration. Variant filtration, the major step, it was performed for variants with a minor allele frequency of less than 1% population databases using a customized AIIMS gene panel. And if the phenotype was matching for a particular variant, Sanger sequencing and segregation analysis was performed. And if a causative variant was not identified, we did homozygosity mapping and copy number variant analysis. So 116 patients were screened during the, families were screened during the study period, and 16 were excluded. 100 families were enrolled. However, 9 had to be excluded from the final analysis, and 91 families were included. In the 91 families, 114 affected individuals were noted. 17 of them were multiplex, that is, I had 2 or more affected individuals. 13 were fetuses, and consanguinity was noted in 21%. So coming to the clinical categorization, 33 families had to be were categorized as syndromic AIIMS, constituting 1/3 of the cohort. 23 and 24 families respectively were categorized as distal arthrogryposis and fetal echinacea deformation sequence respectively. So the overall diagnostic yield was 49.4%, with 47 families having a diagnostic pathogenic or likely pathogenic variant. When we see the category as diagnostic yield, it was variable. In distal arthrogryposis, it was 74%. In FADS neuromuscular category, it was 50%. In pterygium syndromes, it was 45%. And in syndromic AIIMS, it was 39%. So coming to the distal arthrogryposis, 17 out of 23 families had a diagnostic variant, and 10 novel variants were identified in distal arthrogryposis associated genes. One important finding was the only autosomal recessive type of distal arthrogryposis, that is DA type 5D, was most common. It was noted in 8 families. These are the figures of the patients with distal arthrogryposis 5D. They all had a characteristic facial and musculoskeletal phenotype. They had facial asymmetry, unilateral ptosis, and fixed knee extension contractures. In FADS neuromuscular category, in the 14 families where a diagnostic variant was identified, 16 novel variants were identified. This was a notable case of neonate with reduced antenatal movements, multiple contractures, diagnosed with AIIMS type 5. In these 3 adolescents with proximal weakness, peripheral hypotonia, muscle contractures, variants in LAMA2, MYO18B, and COL12A1 were identified. In the pterygium syndromes category, 6 patients out of 11 had a diagnostic variant. 2 novel variants were identified. All of them had variants in CHR and G, that is causative of Escobar type multiple pterygium syndrome. This patient had a blended phenotype. His clinical diagnosis was Escobar syndrome, but on whole exome sequencing, in addition, we identified a likely pathogenic variant in SUS12, causative of Imagawa-Matsumoto syndrome, resulting in a blended phenotype. This is an ultra-rare, clinically recognizable condition, Bartzokas-Papas syndrome, identified in this neonate. In this patient, a variant of uncertain significance was identified in RAPK4. So coming to syndromic AIIMS, there were 13 cases in which a diagnostic variant was identified, and this included 9 novel single nucleotide variants and 1 copy number variant. So this is a very recently described ultra-rare recognizable syndrome, kinship syndrome, with only 7 cases reported worldwide, and this was identified in this baby, a child with global developmental delay, seizures, dysmorphism, mesomelia, and multiple contractures. So in the cases with a normal whole exome sequencing, we went ahead with copy number variant analysis. It was inconclusive, but another important finding was there were 5 patients with a novel phenotype. All of them look similar. They all had developmental delay, facial asymmetry, craniosynostosis, in addition to limb contractures and CNS abnormalities. We are planning to go ahead with trio whole exome sequencing and whole genome sequencing in these patients. So coming to the structural modeling of novel missense variants, for novel missense variants identified in ECL1, RAPK4, and RAPSN, we went ahead with structural modeling. In all these proteins, the structural modeling showed that the variants result either in destabilization, decreased flexibility, or loss of liquid assembly, or leading to loss of protein function. Another interesting finding was 4 patients had 2 genetic diagnoses, 3 of them having 2 conditions causing arthrogryposis, resulting in an overlapping phenotype. So these 3 patients had 2 conditions causing arthrogryposis, and this is the 4th patient who had a SEA1A related developmental epileptic encephalopathy with arthrogryposis, and in addition he had ABCD1 variant causative of ALD. However, the patient expired before the phenotypic manifestations could develop. So coming to the discussion, our study provides insights into the genomic findings of AIIMS and expands their phenotypic and genotypic spectrum. The diagnostic yield, variable diagnostic yield, all were comparable to the previous studies. However, our study included all the clinical categories, and the molecular testing enabled the revision of clinical diagnosis in 4 cases. Hence, this suggests that clinical categorization may have limited accuracy, and hence molecular testing is important. No genotype-phenotype correlations could be identified, and as we see, multilocus variants are not uncommon, and a careful bioinformatic analysis is warranted, and it has implications in genetic counseling. I thank all my patients, their families, and the ICMR funded project for the financial support. Thank you.
Thank you, Dr. Mona. Are there any questions?
Yes.
Please.
Thank you, Dr. Monica, for the work that you have presented. Can you just highlight the implications part of this? So you didn't actually find any clear-cut genotype-phenotype correlation. You did highlight a few cases and some what looked like case series. But apart from that, what would be the implications and recommendations from this work?
So regarding the genotype-phenotype correlations, because our cohort had, when we categorized them into 4 categories, in each category for a particular gene, we had limited number of cases. Especially the most common one was the ECL1 distal arthrogryposis 5D, in which we had 8 patients with the same, in which we could find a phenotypic correlation, but still it was not the same variant, so that we could establish a genotype-phenotype correlation. So maybe if we have a larger sample and more extensive study, we can establish further genotype-phenotype correlations. And the implications of multilocus variants are, this actually reinforced in us that we have to go ahead looking into the bioinformatic analysis further, even if we find one particular variant which could match the phenotype, because some other part of the phenotype can be explained by another variant. If a particular variant cannot explain the entire phenotype in the child. So,
but based on the literature, are there similar data? Because you're limited by the numbers in a specific phenotype and also correspondingly these specific genetic defects. But were you able to do some systematic review or at least the analysis of existing data to see whether there are these correlations?
Sir, genotype-phenotype, actually the large cohorts in arthrogryposis are very lesser, maybe 4 to 5 large studies from predominantly consanguineous Turkish families and not many studies, but maybe if there are particular common ones, then we can go ahead with this.
Okay, thank you.
Thank you, sir.
Right. So, Rajiv, just connecting it to Dr. Lodha's question, are you planning any penetrance studies? Because familial, if we can pick it up early, we can use ART and remove those blastomeres and do it. Is there any implication of that? It could be a very crippling disease, and if we have reached that point, I think we have trajectories which are very promising.
Sir, we had very less autosomal dominant variants. However, when in those in which the parent mother was affected and the child was also affected, we could see complete penetrance. But yes, incomplete penetrance has been known in some. We could not find many in our cohort.
Thank you. Rajiv?
Thank you for your work. Where was the NGS done?
So NGS was actually outsourced, but all the data analysis was done in our
So you got the raw
unit.
data and then analyzed it
Yes,
in-house. Okay, thanks.
Well, so we now come to the next talk. Before that, we welcome Dr. Rajiv Behel, who will be orator for the next session.
So the next session is by Dr. Subranka Datta from Radio Diagnosis and Interventional Radiology. The topic is AIRIB framework, development of an end-to-end artificial intelligence pipeline for the automated detection, characterization, prognostication, and reporting of traumatic rib fracture from CT scans.
Thank you. Good morning, all. I am really excited to present our end-to-end AI pipeline for automatically detecting, characterizing, prognosticating, as well as reporting traumatic rib fractures from CT scans. We developed this framework in collaboration with IIT Jodhpur, and I'm immensely grateful to all my mentors, guides, and collaborators to help us develop this framework, which we envision as a guidance not only for AIIMS but for the Indian academia in general to develop and deploy, most importantly, AI models. So when we were deciding 3 years back to identify a unmet need in radiology, we realized that there are a lot of rib fractures which we often report in our trauma setups. However, because we often prioritize the most important and more life-threatening and severe injuries, we often overlook rib fractures, and almost up to 60 to 70% of our reports, they miss at least one rib fracture. In fact, rib fractures, when multiple, and especially if they contribute to a flail segment, they can cause extended ICU stays and a longer hospital course for our patients. So it's very crucial to identify these rib fractures early, not only for prognostication, but also it often changes the surgical management based on the prognostication risk. There is a 6-point rib score predicting system, which is already validated internationally to predict morbidity risks. However, it's very time consuming. So as you all know, the kind of practice in which we are, we do not get time to report rib score, so it's often limited to research. This is exactly where we realize that AI can help us. Till date, no AI algorithm is there which helps us in computing rib score, and existing AI architectures which can detect fractures in ribs do not have the training to find out those specific characteristics which is required for rib score calculation. And this is the unmet need which formed the inspiration for our current study. Our objective was to generate an annotated and expert-reviewed dataset of thoracic CT scans with rib fractures. We evaluated all the existing machine learning architectures, based on which we developed our novel model. And finally, which was the most ambitious part of our project, was to develop a pipeline for automatically generating a structured preliminary radiology report right from the DICOM. So this was one of the first studies, and we wanted to demonstrate feasibility. So we developed a pilot IRIB1 dataset in which we took 3 different CT scanner models to ensure heterogeneity and applicability to different setups. We annotated and verified each fracture on all the slices of the CT scans. And finally, for the machine learning model training, we had a training set where we taught the computer to basically figure out where the fractures are. And we had a validation set to fine-tune the hyperparameters of the model. And finally, we tested it on a testing set to find out how accurate and how good our model was. We evaluated all the existing machine learning architectures on the IRIB1 dataset. And finally, we chose, I won't go into the technical jargon much, but UNet is a very good and state-of-the-art model for most of the segmentation tasks, which essentially detects where exactly the rib fracture is. And we explored various adaptations, and we also came up with our own novel adaptations after brainstorming with the technological people, which was the Model 5, or the Dia Unit++, wherein we used a morphological dilatation operation to make our finer segmentations even more robust. For the classification, we used an architecture called the ResNet to categorize and classify all the rib fractures. So what did we find out? Usually in machine learning, we have 2 scores, sensitivity and dice score. We found out that our novel model, which was developed after brainstorming with the engineering team as well as the clinical team, had the best dice score of around 40.17%. So to demonstrate the kind of outputs which we are getting from the models, on the left-hand top, A is the rib fracture, which you can see in 3 different planes. B is the one which was annotated by us and reviewed by a radiologist who is an expert in trauma. C and D show the segmentation masked in white. So the green part is the ground truth, which we had manually segmented. The white is the output of the two different machine learning models, and you can see that it is adequately covering the entire fracture area. For the classification task, the fracture characterization, which tells us whether the fracture is displaced severely or an undisplaced buccal fracture, that showed the highest area under the curve. Finally, we have the prediction of the RIP score, wherein each fracture gets assigned a particular score according to the specific characteristics depending on the displacement, whether it's contributing to a flail chest or not, and we give a final prognosticating score for each patient, which is called the RIP score. To summarize, our Dia unit ++ model showed the best sensitivity and dye score. The ResNet achieved the best overall area under the curve and sensitivity for the classification. So we envision IRIF framework as a road to clinical deployment of AI models in the country. So we essentially will be just feeding in the DICOM file or the CT scan. It goes through our Dia unit plus segmentation model for finding out the fracture. The ResNet classifies the different characteristics. And finally, a RIP score is assigned to each patient, which helps us in prognosticating. And ultimately, the most ambitious part was the creation of an automatic textual report. And on the left-hand side, you can see this is currently how we report RIP fractures as a part of the thoracic CT trauma report. But when we feed it to our pipeline, this is the very detailed structure reporting in which the clinician, the trauma surgeon, knows exactly what the RIP fractures are in a patient and whether there's a frail segment or not, and I can also prognosticate the patient. To conclude, our pilot study was one of the world's first pioneering efforts in trauma radiology for automatically prognosticating using a validated scoring system. It not only helps us in reducing the number of missed fractures, which has significant medico-legal implications, but also helps us in prioritizing our CT scan based on the RIP score. So if a patient has multiple RIP fractures and a flail segment, he has a higher RIP score, and we can tell the trauma surgeon, please look at this patient earlier. It also streamlines reporting. We all know across all health systems in the US, NHS, even in India, the reporting time is significant. So it helps us in reducing the reporting turnaround times. We presented our work in the RSNA last month, and it won the international award for the best paper for research trainee. And also it is now considered among the top 10 machine learning architectures on ANPINI internationally. Thank you.
So, any questions from the audience? Okay. Yes. Please stand up.
So thank you. So I was just kind of wondering that, like it's a great study, but, in a study set, you have a quality assurance. But when you are going to suggest this algorithm is going to be used everywhere, how does the quality assurance of radiology reporting or the scanning quality, does it matter or not?
Right. So you can give your answer by also enveloping another query that you rightly mentioned initially that our clinical workload is very exhaustive. Right. And we are able to pick up things which are run-of-the-mill and middle-of-the-road things, but outlier things get missed out. The reporting will become very easy. You will have a very specific objective report so that the priorities can be set in.
Right.
But the outliers which are missed out, they had to be fed into the system. Otherwise, AI will not do anything. That's the limitation. At the same time, how important clinically are these outliers? Because you will get so much of work then, and the number of people who would actually deliver will be fall short. They can together answer this.
So the major problem to clinical deployment of AI tools, sir, is that there's a huge amount of bias which gets created. So once we start relying on these tools, often these outliers which you mentioned are small fractures which might get missed. So how the US system works is that they give a basic training to our radiologists to essentially understand the kind of biases in each deployment strategy which their system is taking. So once we are deploying this in the clinical practice, there are certain kinds of RIP fractures which might be missed unless we are creating these outliers in the second version. So this is a preliminary work. We have already been feeding more outliers which are missing to retrain and to account for the drift of the data sets to make it more robust. And so essentially it's education of the clinician and not over-reliance on the outputs of these, but to also basically use this to help you in reporting so that your time is saved.
Okay,
Yes,
so Dr. Datta, thank you. I think that was very interesting work.
Thank you, sir.
A couple of questions, so while you used the dice score to say that your model works well, I think in very simple terms, if you could tell us of 1,000 odd fractures, how many were actually identified by a model?
Right. So that is the sensitivity, sir, essentially. And
of the tool, you talked about sensitivity of some other parameters. I just was, what's the sensitivity and specificity of the primary detection tool?
right, so sensitivity was around 70%.
Okay, so
This is for pilot study,
70%
yeah.
is something which you still would, there's scope for improvement.
Definitely, sir, yeah.
The other thing is that, you mentioned that clinical, let's say, when you're routinely reading, you, let's say, tend to miss these fractures. So what measures you put in place to make sure that the gold standard is actually gold,
right,
and not, let's say, there are issues because your final sensitivity would then actually be much lower than the 70% that you found if there were some issues there.
Yes, so how we tackle this is essentially, we had the reports, which we had initially, but we had one radiology resident to go through all the scans we annotated. Then we made it reviewed by an expert trauma radiologist who went through the scan. So how we work in our setup is initial scanning and reporting is done by a resident.
I think that's okay. So
yeah.
from the study perspective,
yes,
how did you ensure that there was this system worked well?
Okay, sir.
You could have reporting which is like you could agree.
Right.
How often you found discordance? I guess some of those things are also important to highlight actually what the system may do in practice, because 70% is something which may be good from a usual reading, but it may not be great in terms of an overall sensitivity which you're looking for a tool.
Yes, sir. So this sensitivity was from like around 5 months back. Currently, we are using spatial temporal coefficients, so it is much better. So this is a pilot study with only 100 scans. So the project which we are doing is for 1,000 scans. So that automatically pumps it up. We are just trying to find out our own novel model, whether it can beat the state of the art, which it did on our data set for now.
Okay, fine. I think the measures that you use are often not what would be, let's say, directly have reading or implication clinically. I think clinically you would be looking at sensitivity, and would have been good to see data on sensitivity of various models as well. Maybe I think we'll just discuss that later.
I had the data actually, it's around 70%. I had mentioned.
For all these three?
So
for
in
all the
interest
models?
of time, please excuse me, we will close the session now. The questions can be done at the later stage.
Thank you. Thank you.
Thank you.
Thank you very much.
Thank you, chairpersons. Thank you, judges. Thank you, speakers. And now I hand over the stage to Dr. Vivek Dikshit and Dr. Sunita.
नमस्कार। तृतीय अनुसंधान दिवस के आज के इस गोल्डन जुबली ओरेशन सत्र में आप सभी का पुनः बहुत स्वागत और अभिनंदन है। आज के इस सत्र के लिए मैं निवेदन करूंगा डॉक्टर के वर्मा, डीन एकेडमिक्स, डॉक्टर JS टिटियाल, डीन रिसर्च, डॉक्टर गिरिजारथ, रजिस्ट्रार एम्स, और आज के सत्र के मुख्य वक्ता डॉक्टर राजीव पहल, DG ICMR and Secretary DHR to kindly come on the stage.
I now request Dr. Girjarath to please welcome Dr. Rajiv Bell.
माननीय निदेशक महोदय से निवेदन है कि वे कृपया मंच की ओर आगमन करें।
So please, would you like to introduce our speaker of the day?
Dr. Rajiv Bahel, the DG ICMR, Dr. K.K. Burma, the Dean Academics of our institute, Dr. Jaya, the Dean Research of our institute, faculty colleagues, residents, students, ladies and gentlemen, a very good afternoon to all of you. On behalf of the institute, I welcome you all for this fourth edition of AIIMS Golden Jubilee Oration. So the previous speakers for this oration have been Dr. Balram Iron from AIIMS Delhi, Dr. Saji Kumar from Mayo Clinic USA, and Dr. Rakesh Agrawal from JIPMAR Pondicherry. So that is why this is the fourth edition. To carry forward with the proceedings, now I invite Professor K.K. Burma, the Dean Academics, to introduce the speaker, please. Professor Burma, please.
Thank you, Dr. Rath. Good afternoon, ladies and gentlemen. I have the honor and unique privilege of introducing Dr. Rajiv Bahel, who is the Secretary to Government of India, Department of Health Research, and Director General of Indian Council of Medical Research. I don't think this audience or any scientist, researcher, or healthcare professional needs any introduction of Dr. Rajiv Bahel. But just to highlight some of his contributions, the achievements, and the credentials that he has made in the field of research, to let you know that he is a pediatrician by training. He did his pediatrics training at University of Delhi, and then he got his Ph.D. from our own institution, from the Department of Pediatrics. I was just talking to him, when did he finish, and then what did he do after this? So he stayed here till 2002, and after that he moved to WHO. And in WHO he stayed for 20 long years in different leading positions, guiding, monitoring, and supervising research. And his research areas primarily focused on nutrition, diarrheal diseases, and then, you know, a person of his caliber, his stature, doesn't have just one thing or maybe just a few things to do, but there are many. Something like he has been doing the conceptualization of research, setting the research agendas, implementing research to address the key priorities, mobilizing resources, supporting and building capacity of research institutions to implement research studies with excellence, and translating research finding into public policy and programs for the larger public good. In addition, he has significant contribution in enteric and respiratory diseases and non-communicable diseases also. He has conducted several and coordinated many population-based and hospital-based interventional trials, cohort studies, and implemented research to scale up interventions. So his research has not been limited to these areas. He moved on to the policy planning and then the program implementation, and focused on, say, the public interest research, how this can be translated into larger good. he has led research studies in over 20 countries and strengthened research capacity in over 50 institutions across the world. Some examples of his research contributions which have made a lasting and larger impact on the public has been something like use of zinc in the treatment of diarrhea, early initiation of breastfeeding, outpatient care of newborn with severe infection closer to home, which included intervention by frontline health workers with simplified antibiotic regimes, and the initiative of kangaroo mother care. That's very well recognized contribution that he made in the field of research in his own specialty. So we are truly privileged to have Dr. Val Yu with us here today on this special occasion of our research day, where he would be delivering the Golden Jubilee Oration, the very befitting oration to him, and talk about medical research in India, vision and action. So welcome to Dr. Val once again, and I hand over the now stage to Dr. Val. Thank you.
Thank you very much for that kind introduction. When I was asked, and I couldn't say no to Dr. Srinivas, Dr. Vineet Ahuja, and others, that I should come here and talk to you all on this day, I thought, what would I say? Should I tell you about the research I have done? A lot of that has been said. Or the other option was, should I share my thinking about research in medical institutions in India, of which the All India Institute of Medical Sciences is the first, most important maybe institution. But broadly, should I tell you more about what we are thinking, what I am doing? And I thought maybe the latter would be of some more interest to you, but more importantly, it is something that I had promised myself, that I am not going to talk about myself. I'm not going to go and talk about work that I have done in the past. I will focus these 3 years that I have as DG ICMR on trying to do things that the Indian Council of Medical Research, how it can help the country improve research. So I'm going to generally talk more about, not about myself at all, but more on my work. I'm going to talk about what I'm doing at the moment. Firstly, I would like to start by congratulating you on celebrating this Research Day. It is so heartening to see this whole hall full of people in such an important day that you have given it that importance that you are celebrating it the third time as a research day. So this is really very heartening to see, and I congratulate you, Director Sir, and others for that. What I'm going to talk about is something very simple. What are we trying to do? What is my vision? What is the vision of the country? What is the vision of the Government of India, where our Honorable Prime Minister has added Jaya Anusandhan to Jai Jawan, Jai Kisan, and Jai Vigyan? The vision is that the Indian institutions will achieve global leadership in health research, but not just that. It will also help solve health problems of Indian citizens. But where are we now? Are we in the global leadership position? Well, hard to say, but there is one ranking called Schimago ranking, and I'm only looking at ranking of medicine. Year 2021, I didn't choose 2023 because they dropped one of the institutes in 2023 for some reason. And here are the top 5 institutes in the country. The first one in 2021 was All India Institute of Medical Sciences at rank 396 in the world. Today, 23, it is still around 285. And the others are significantly behind, but PGI Chandigarh, Manipal, Banaras Hindu University, CMC Vellore, and others. There are no institutions, obviously, in the first 100. So that is the first target, that we should at least have 3 Indian institutions in the first 100 in the next 3 years. It's really hard to go there, but I'll talk about some things that might help. Just to note that what is this ranking made of? It is made of 3 things. One is research, which is citations, impact, publications, etc. But it's only 50%. The remaining 50% comes from whether that research leads to innovation. Innovations in terms of patents, technologies, something, products that have come out of that research contribute 30%. And the remaining 20% is societal interest in research. So they just scan social media, websites, newspapers, etc., and see how much is the interest in research. Now, clearly this has increased in the recent times after the COVID pandemic. But we should keep in mind that publications, even very good publications, is only half the work done. The remaining half is to convert that into something that is used by people, and then it also gets people interested in that and reporting that. So keep that in mind. If we have to be, there were almost no institutions earlier other than the European, American, obviously USA is first, 89 or 88 out of 100 are from US. But you will be surprised to know that 11 are from China now. And they are going to increase in the top 100. So what are we going to do? We are looking at the people who can change this. You all, you are number 1 in the country, but you are still about 300th in the world. Also, on individual basis, scientists. So this is the Stanford AD scientist ranking. And I tried to look into it for medicine and health. And I found about 28 Indian medical scientists are currently in top 2%, which looks very good. It's a large number, unless and until you start comparing it with other countries. When you look at United States, it's not exactly the AD ranking, but more or less half of the people are from United States. So we are far below, again, we are not going to be even in the first 10 or 15 or 20 countries. So the question is, can we increase these numbers? If not to 500, but at least to 100 medical scientists in top 2% in the next 3 years. So that is the vision about global leadership in medical and health research. But in terms of solving problems of India, in communicable diseases, our biggest problems are to currently eliminate TB, malaria, filariasis, Kala-Azar, leprosy. Have you seen any developed country where TB and malaria and leprosy are huge problems? And we just cannot get to this 2047 as Viksit Bharat if we cannot eliminate these diseases. And they will not be eliminated unless we work on research and innovations. Because nobody else in the world will be working on them. Those days are not their problems. Only a few people are interested from the Western countries in these issues. These are our problems and we should solve them, and we will. In terms of non-communicable diseases, the growing burden of NCDs, diabetes, hypertension, we need absolutely great research to prevent and manage non-communicable diseases our way, not their way. Because if we start treating with drugs and medicines all the 100 million people with diabetes in India, we will go bankrupt as a country. There are mental health problems and aging problems, and all these are increasing. As we are growing older, we are getting these, and this is key to when you think of research, when you write your research projects, don't lose sight of these issues. We need to greatly reduce neonatal mortality, childhood malnutrition, anemia as core problems of child health and nutrition. How will we do that? So, 3 possibilities: develop new drugs, vaccines, devices, and diagnostics. Now, you may or may not be able to develop them by yourselves, but that is why we have created, and previously, right from the time DBT, ICMR has been creating these ICMR, AIIMS and ICMR advanced centers or centers of excellence between medical institutions and our technology institutions, so that we can get the research into development. So unless we are able to get rapid indigenous development of affordable technologies like drugs, vaccines, future-ready devices, diagnostics, we cannot again get the access of these to all the people in India. Improving quality of health care through interventions that improve clinical care at primary, secondary, and tertiary levels is something that clinical researchers can think about and public health researchers should think about. Again, implementation research, health technology assessment to make affordable healthcare technology to everybody is some other people who are interested in that type of research would be very important to move towards. So that's where we want to go. So what are the strategic actions? I'll talk about a few from ICMR. Then I will talk about what the institute can do, and in this case AIIMS. And then I will talk about what individual scientists, younger and not so young, can do to change what we are, how we are getting and making progress. So 5 things that I will talk about is what ICMR is doing in the area of enhancing research capacity and what is new, reform of the extramural grants program, strengthening of the research ecosystem, a highway for medtech product development, and greater use of evidence in policy. So just to show you a few things about capacity development, fellowships, and grants. In terms of numbers, the MBBS STS fellowship, we have now fixed to 2% of all medical seats. As the medical seats will grow, so this number will grow. And MD-MS thesis as well to about 1%, so it is a 4-fold increase in the MD-MS thesis grants or the awards. The important one that I want to talk about a little bit is the MD PhD ICMR fellowship. So it used to be about 25 fellowships that we would give as salary to those who have a medical degree, an MD, to do a PhD. However, there were very few takers, almost none. So we have thought of changing this to a different type of fellowship. And we will start with the faculty of medical colleges and medical schools and medical institutions. All the young faculty who are interested in doing a PhD, we would encourage them to apply, and they will be given a grant of 30 lakh rupees. We are at the moment 30, we can even increase to 50 lakh rupees to do their first big research project, in which they will also be able to do their PhD. So all they need is to find a good mentor, a guide within their own institution, and maybe one co-guide in ICMR, if possible, if they are going to register in ICMR. For AIIMS, this is not a requirement, because you can register for PhD within AIIMS. So those MDs, who particularly those who are in clinical subjects, and they want to get initiated in research, they can enroll for a PhD program within their own institution, and they will get a grant for doing their PhD research project. Why only not just a grant, but also a full package of research training that you can do at your own leisure, with your own time, whether it is your vacation or weekends or whatever, all online courses, but getting about 7 or 8 key issues in research methods and research learning. Our aim is that if we start getting physician scientists interested in research early on in their career, let's say even below 45 years of age, then they will be able to get a little more interested in research. A few more percentage will get interested in research. We have increased the number of the PhD fellowships from 150 to 300 for MSc PhDs, and also added 200 non-medical fellowships for PhDs within INIs, of which you are a premier one. Again, this is something that you can take advantage of. We have now 300 grants for non-ICMR scientists to travel, including international grants, as well as for workshops. Previously they were as is, now we've put a very large number. All this is meant to create more interest and more capacity in medical research. I'm also very delighted about that ICMR has now become, in partnership with the CSIR, Council for Scientific and Industrial Research, their Academy of Scientific and Innovative Research, and ICMR has jointly opened a faculty for medical research on 13th of December last year. And the Office of Dean will be at our National Institute of Malaria Research campus in Dwarka, and we will be able to award PhD and master's degrees for our institutes, but also to any other institute which does not have their own PhD. So whether it is an MD PhD or an MSc PhD, if any medical college in the country the people want to do a PhD, whether they are MSc scientists or they are MDs, we should be able to enroll them in the ACSIR ICMR Faculty of Medical Research. We are also going to start a bunch of niche master's programs. Now, when I look at the faculty, it's on the right side, I talked about MD PhDs. Just look at the numbers in the United States, which leads the world in the MD PhDs. So there are about 12,000 MDs who are also PhDs in the United States. I don't need to tell you the number in India. They are really small number. Another initiative that I want to talk about a little bit is the postgraduate thesis. Dr. Paul was telling me yesterday that when he was at what was erstwhile MCI, now NMC, in the middle it was something where it was BOG or something. He wanted to abolish thesis of MD students, saying that what a waste. Most of the people learn to cheat, don't do serious work all over the country, not necessarily in AIIMS. However, I disagree a little bit with him that we should abolish it. It does give people some research training. And the more interesting thing would be to try and make it better. And one step we have taken is that we have put together groups of good guides across the country and asked them to make topics for thesis that are good for students at all levels, whether they are in AIIMS or in other state medical colleges, etc. And those we have made available along with National Medical Commission for students to work on that and their guides to work on those thesis topics. And also like CTRI, create a registry of these MD, MS theses, including maybe even DM thesis at some point in time. Because if you think about even the DM program, if you go to any other Western country, the fellowship program is at least 50% research and a lot of basic research included in that. Whereas if we look at our DM, it is basically the next level of MD, essentially clinical care, a higher level clinical care. On the side of research funding, just to know three things that have changed. One is that the small grant is not so small anymore. ICMR used to give grants of average size about 30 lakhs. Last year, the average size was 70 lakhs. And the cap is at 2 crores. So the small grant is up to 2 crores. This matches anywhere in the world, $250,000 US is a small grant anywhere in the world. And we are not going to give a grant which is less than 10 lakh rupees. So 10 lakhs to 2 crores. And we funded 600 last year. A new grant was created, which many of you know about. Many of you have got it as well. Congratulations. AIIMS Delhi gets about 12% of all ICMR grants. Last year they got 12% of small grants, similar percent of intermediate grants. The Centers for Advanced Research, I won't tell you immediately, you will know in a few days. But all these are at a much higher level of funding. The intermediate grant did not exist in the past. It was only task force and that had to be managed by ICMR. This is investigator initiated grant up to 8 crores or $1 million US. Average this year was 3.5 crores. A little over 100 grants were given. The Centers for Advanced Research averaging at 13 crores, about 30 grants will be given, give or take 1 or 2, depending upon how the final committee decisions are made. And in addition, we have launched a national research priorities program in which 15 projects, multi-state projects, multi-district projects have been started in different areas of our priority, including those that I showed you in terms of communicable disease, non-communicable disease, and reproductive child health and nutrition. Also a piece of good news about our research budget in the ICMR. You can see the sizes of the blue bars. The orange bars are the same but adjusted for inflation. So they are mostly in dollar terms. Whichever way you look at, there has been substantial growth from 2015-16, and what increased during COVID, we were able to not only maintain that because there was a talk about reducing the budget, we managed to increase from those 13. These are only money for grants that ICMR gives, not our institutes, not salaries, not others, to 1,600 crores this year. Another shift in the program has been that earlier in the past years, most of the focus was around descriptive research, just trying to find out, describing the problem, describing the associations, what a genome looks like, etc., etc. However, this year you can see that between description, discovery, development, and delivery, there is a more wholesome balance. For small grants, descriptive research was only 22%, discovery 29%. I would say some of that might be just description in the hope of discovering something, but still 42% development of new products, 7% delivery, and similarly intermediate grants, much more on the development side, and the centers for advanced research as well. Another thing which we are doing is the multidisciplinary research units. For AIIMS, that is not so critical. I will not go into great detail, but we have more than 115 of them in different parts of the country. And it is not only INIs that are important, but also what research is done in those institutes. Even if they do small number of projects, they can add to India being a force. The last two things about ICMR is we have created an innovation ecosystem called the MedTech Mitra, which is hand-holding medtech innovators, which means whether it is diagnostics, vaccines, therapeutics, devices, for clinical evaluation, regulatory facilitation, and uptake of new products. So this is only for those products, those things that require regulatory approval. So you know very well these valleys of death between the time that you decide what is the need to getting a proof of concept, to a prototype, to a product, and to the scale up and public access. So we have partnered with primarily with CDSCO or the Drug Controller General of India to provide a regulatory pathway and support to anybody who is going through this challenge. So starting with fostering what I had talked about earlier, the engineer-clinician partnership that ICMR does, along with Atal Innovation Mission, the pre-compliance gap analysis and testing done by our partner called AMTZ or Andhra MedTech Zone, particularly for devices, and the regulatory strategy, which is the most critical piece of advice that we can give to the preclinical and clinical evaluations that will be funded by ICMR when we see that a product is promising. And this is beyond those extramural in small, intermediate, and CAR grants. And then finally, through guidelines and health technology assessment, taking this to scale so that once approved, these can be done. So this whole pathway has been put into action. More than 100 innovators have already come to this portal, and about 24 have already been counseled as to what the next steps should be. There is no point in creating evidence if it is not used. In DHR, we have created a Center for Evidence for Guidelines and made an agreement that all the Ministry of Health guidelines will be evidence-based and will be done in partnership between the Department of Health and Family Welfare, Department of Health Research, and the DGHS. And health technology assessment, which is the cost-effectiveness assessment for all the health technologies, has now been made into a permanent office in DHR. So these are the 5 things that we are doing from our side. What do we ask, or at least my thoughts at 3 a.m. this morning when I was trying to make these slides, that what should Dr. Srinivas do? You already have an intramural grants program, but many institutes don't. You have, but you could expand and enhance your intramural grants program. We have also discussed between Dr. Srinivas and I that we will create a ICMR AIIMS partnership for at least a few top priority projects where we will decide the priorities together and then fund something which we think will be yielding something useful. We'll start with a modest amount of 25 crores next year on that issue. Greater value to impactful research. So we give value to research, surely, but that is largely for promotions, getting jobs, looking at impact factor, looking at publications. But people are moving away from that, and I started by saying not only do we want to become a global leader, but we want to do research that has impact. So when you are starting from the fact what you give to your MBBS student or to your MD or MS student, even if it is simple, you don't need to have very complicated next genome sequence necessarily type of research for every project, but give them something that is useful. For me, that was transformative. When I joined Dr. Bhan in AIIMS in 1994, the first project that I did was a project in which WHO would decide based on the results of that project whether there would be global EPI-linked vitamin A supplementation or not. And believe me, that completely changed my view of research and the way I prepared, the way I took not only that project, but the rest of my career. Because I knew the consequences and the implications of my study were so important that I was part of. When we say these are just students, we are just building their capacity, I ask, what are you building their capacity in? They are just PhD students. So what? You see anywhere in the world in a developed country, the cutting edge of research is the PhD student. Why are we wasting and teaching them mediocre research, which has just very little value? So I am saying give more value to impactful research, not just people have publications or research projects. Another very difficult ask, protected time for faculty who is engaged in impactful research, which basically is a very hard deal, but until we do that, essentially we are basically making people say that those who work hard get more work, and they continue to do everything else, but they also do this, which all of you are doing, which is absolutely great, but imagine what you could do with a little protection of time. Central research lab for research, huge. People should be, there are so much, there is equipment that is not used enough. Everybody wants their own. Why can't we create central instrumentation facilities, central research facilities. And I am sure Dr. Srinivas has already told me he's thinking about that. Another important issue is ease of doing research. Again, with the research section, I am really proud of AIIMS to have a research section which is so active. But there is a constant need to keep thinking how we can engage more people and make it easier for them to do research. And you also have taken a lead on the entrepreneurship policy. If we could operationalize that policy and where people can see some people make a lot of money coming out of this, it will probably encourage others. My last segment is the strategic actions for most of you. This was for Dr. Srinivas and the leadership. What can you do? So, people who are not in this room, faculty in particular, how can we bring them here so that they get a greater proportion of faculty is interested in conducting impactful research? When you are writing grants, focus on solutions. Do not just describe the problem unless the problem is so unknown that you must describe it. But at least try to take that. If you do your first project as a description, the next problem should try and find a solution to that problem, even if it is difficult. High impact publications that affect clinical or public health practice, I've spoken about that. International collaborations, don't sell yourself short. At HMSC, when you come for approval, this is one of the things I have instituted, personally said that we should have it, is how is the international collaboration treating our scientists? Are we being treated at par? Are you getting authorships that make sense, particularly if you are the main site? If you are one of the 50, I don't ask you to be necessarily the first, last, or corresponding. But many times we lose out because we think, okay, we have something, at least we have some collaborative grant. We should try to do more, but to get those authorships, the corollary is you have to conceptualize, you have to be part of the conceptualization, grant development, protocol writing. If you come in later, these are all taken by the time the collaboration comes to you. So seek collaboration early in the development of the project. Partner with industry with your entrepreneurship policy. Collaborate, and collaborate. If there is one secret sauce that is missing in our country, I have been to so many in the world, I have seen that most developed countries that do well are able to collaborate even with people that they don't like. We are not able to collaborate even with people we like. And more so, the closest ones to us within our own departments, next door neighbors, etc. It is a mindset. When I have time, I will prove to you through my own personal experience. I can tell you that you will gain more in collaboration than those who go alone. It will always be true, and it is a fact. No matter what you think, in the long run, you will gain more if you are able to collaborate. Two small things. Cite relevant Indian publications. The first thing you want to cite is somebody somewhere has done in the Western world and published in the Lancet. Why not in Indian Pediatrics or Indian Microbiology Journal or whatever? Why don't we cite, we publish thousands of papers. So citations and impact comes from this. If we cite other people's work, and you can do that consciously. Promote Indian publications through social media, yours and others, because even that has value. because it generates not only public interest, but also global leadership. So this is in general. You have role models, and this is my own list, so please don't. These are the ones on the left side that I have worked more closely with, and on the right side that I have seen as my role models in the past. They are not by no means the only ones. But these people from this institution have shown the way what you can do, whether it is Professor Bhan, Professor Reddy, I.C. Verma in medical genetics, Professor Paul, and of course Dr. Ramalinga Swami, Dr. Paintal, Dr. Tandon, Dr. B.N. Tandon, Dr. Pandey, and many, many others. This whole page could be filled. I just happens to be my favorite list. Everybody would have theirs. And one of the last messages, what I was thinking, what will I tell the young people? What I'm telling you what Dr. Bhan told me. He told me, you are not a complete researcher, and particularly me, because I didn't do clinical work, I was doing only research. He said, unless you master all these 10 skills, methods. Methods is the break point by which your good idea will get funded or not get funded, published or not get published. Good results versus good means believable results or not believable, credible results. Quantitative and qualitative when you require. Critical thinking, conceptualization, grant proposal writing, protocol writing. You may think I'm not a lab researcher, but Dr. Bhan made me do microbiology, immunology, biotechnology, biochemistry, including atomic absorption spectrophotometry, thousands of samples with my own hands, at least once, so that you know what you are dealing with, you understand that, and then you may not do it ever again. Bioethics, rigorous study implementation. It's not that I have hired my coordinator, now they will do the research. I gave the idea, I have no other time. That is not how good quality research is done. Again, quoting Dr. Bhan, my own mentor, said, great idea, world-class implementation. Both go together. If you do the two, you have success. If you don't do either one of them, it's B-. If you want to be A+, great idea, world-class implementation. Data management, data analysis and interpretation. Sometimes people think this is biostatistician's role. Every researcher, if you really want to be, in Dr. Paul's words yesterday when he was traveling with me, he says, oh, the complete Raymond man, or in the ad that used to come. You need to have these skills, if not all 10, as many as possible. Systematic reviews, meta-analysis. Even if you do one, even if you are essentially not a secondary researcher, or understanding and working with public health programs, engaging with communities for research, costing, cost effectiveness. These are 10 skills that either you should have or you should form a team that together has each one of those skills. But at least try to learn something about each one if you are young. If you have the time, learn to do these, and that will take you very, very long way. Just want to say one thing about, because I thought the last slide which my OSD had made, so I said I'll tell you this. I've resigned 3 times in my life. First time I had a set career. I was a second-year senior resident in an institution where I was also the best postgraduate And almost everybody thought I'll get what I wanted to be, a faculty, clinical faculty in Kalavati Saran Children's Hospital. I resigned that job to become a research officer with half the salary and no future prospect in 1994. The second time I resigned was when I was a permanent scientist in AIIMS. But I thought, I've done 10 years, I want to do something else. So for an 11-month job in WHO, that was the second resignation. My third resignation was when I had 9 years still of service left in WHO with a great job, which I just did last year, and I resigned the third time. Essentially, what I'm saying is, I tried to follow my heart, not my brain, and took the path that I thought I should take. So, in words of Robert Frost, he says, 2 roads diverged in a wood. I took the one less traveled by, and that has made all the difference. I'm not saying that you have to do something completely different or completely crazy. However, please do take risks. Please do take the road less traveled sometimes, because it can take you somewhere where no one has ever been before. Thank you very much for your very patient listening. Jai Hind.
Thank you, sir, for that thought-provoking and insightful talk. I now request the chairpersons to please felicitate Dr. Rajiv Bhel. I also request the AIIMS Director, Dr. M. Srinivas, to join us on the stage for the felicitation of Dr. Bhel.
Thank you. I request all the dignitaries to please remain on the dais for the next session. I request Dr. Archana and Dr. Tony to take us for the next session.
It is time now for the AIIMS Research Excellence Awards. We will commence this session with the lamp lighting ceremony. I request all the dignitaries on stage to please proceed with the lamp lighting.
Thank you. With that auspicious beginning, I request Professor Jaya, the Dean Research, to present his report. Sir.
A very good afternoon, friends. Welcome to this AIIMS Research Day today. Today's Golden Jubilee orator, Dr. Bahal, Director AIIMS, Dr. Srinivas, Dean Academics, Dr. Kaushal Verma, Dr. Girija, Registrar, the previous Deans of Institute, and senior faculty, my faculty colleagues, students, and dear friends. Indeed, it's my pleasure to deliver this research activities which we have done last year and talk something about which we have to achieve and what we have done in last year. Let me begin with the actual research we've done last year, extramural projects. I think this highlights what Dr. Bahal talked about. If you look into last year, we had almost 863 projects being done, and the largest segment is ICMR supported research. He said 12% of their research segment is by AIIMS. We want to make it that almost 50%, sir, and you have to give enough support to AIIMS for getting more projects to our students and faculty. And looking into this, one important area, if you look into the second last is international projects, which is hardly 59. I think national importance, Institute of National Importance, this segment has to be in hundreds, and that will make us reach some step forward, which Dr. Bahal talked about. AIIMS should be amongst the first 100 of research-related institutes. For that, this amount of 126 crore should translate into 1,000 crores in the next few years. And that is how we'll have an impactful research and some important things coming up in terms of innovations, patents, and which will translate to actual patient care for their country also. And second important segment for us is the intramural research segment, which we have been doing for a large number of years, and that holds a significant amount of research being done by our students and younger faculty also. There also, in last year, we had 3 important segments added in this. One is a Faculty of Nursing Intramural Project Scheme, which already started. We have 6 projects there. And other importantly, the financial support for a MD, MS, MDS, DM, MCS thesis. And that is, I think, one of the important segments for young people to look into how they can have a better research-related thesis. And one of the challenges have been some sort of financial support for these research work. Once we give more amount of money to these people, definitely we'll have more better research in this area also. ICMR is doing great job in this, and I request the AIIMS administration also to look into that. Maybe you can increase some amount here also. Other area is we are looking for a AIIMS IGIB interdisciplinary collaborative research, and that's in process. That will also increase the interdepartmental research also in this area. The third area of research section has been to give a lot of funding for international travel, and that is from the AIIMS Endowment Fund. In 2022-23, we had large segment of people getting this fund from International Travel Fellowship for undergraduates, PhD students, staffs, from the Infosys National Travel Grant for UG students. Other concern have been to have some significant amounts of endowment fund generation, like we have Shobha said, Wagner Chair for gynecology, oncology areas. Similarly, we are looking for more donation in this area so that we can have more chairs, more amount of research being done in this area also. And that is going to be a more important thing also. As you all know, the research section is not a small area. It's a big department, I can say. And thanks to the AIIMS, generation of AIIMS think tank, they have built this area. And you all understand research has been one of the important motto behind the development of AIIMS as a national importance institute of this country. And to be very frank, we have achieved that also in last 75 years. AIIMS is one of the leading medical facilities, not only for patient care, teaching, also for research also now. So we do have CCRF, and that's going to be one of the important area for younger people to get into that. We have developed the Center for Medical Innovation and Entrepreneurship last year, and clinical research unit, and we already had intellectual property right, technology transfer division working for last year or so. CCRF is, I think, is one of the biggest thing which we achieved in the last few years in center, to create a centralized institution where you can have all the research facilities in one particular wing, and all the people who have a research projects, research mind, right from the student to a scientist, can avail that research. And it was rededicated to AIIMS in 2021 by a Nobel laureate, Professor David Julius. And this will really give access to all people to do our research in a comfortable zone. We already have 12 scientists been recruited in this area to support you in these areas also. We also do a capacity building workshops, training program. This was the last year programs. And other thing which we added was central clinical unit. And this is been a good unit for all of us, not only for our clinical people to do a research. You can have a lot of things, IIT-based research can also be done in this. And Dr. Mathur is looking after this. Dr. Vijay Mathur, he is the faculty head of this unit. And we want to do a lot of more activities through this, invite all young people to get into this area also, involve into workshops, which is going to train you how to do a good ethical research and come out with a better innovative ideas also. This is one area which is going to be a future for our research, not only for AIIMS, for all the medical institutions also. And Professor Alok Thakkar is the faculty head of this particular area, and he's doing a wonderful work. And I wish that it goes further and makes big impact on a national front and international front also. And this is what area where I think we had thinking to how to make people more comfortable getting their research patented, so that we have a better regulations and have more better research coming through this also. And this area also would be a future area for us to develop, to make it more innovative, more entrepreneurship coming in this area also. This is a recent update from our research section. We signed the MOU with an inauguration of CWD, where we have the now access to central census records for us, where people who are looking for a community-based research can also access the data, and we'll have a very helpful data to generate, and access to that data will create more impact on research also. So we have already looked into how to make this data applicable to our people looking for secrecy in terms of not letting this data leak out to other people. And we have data from 2001 to 2011 census for us, and the next census will give more data for us also. These are various WHO collaborative centers with AIIMS, and that is also a very important segment for us, because this WHO collaboration gives you some sort of a realization that institute is doing good work. Apart from that, gives you a lot more other areas, not only for research, it also gives you access for a development of various programs and getting into the basic areas of our concern, also in all health sector of our country. These are association we have, name few, we have IIT Delhi, Kharagpur, then you have TSTI, IGIB, and the triple ID, which we have been associated now. And we require more such collaboration, not nationally, internationally also, to develop area of research which is bigger than people can think for future also. I can see the young people sitting here, the future of research with them. And this is the team we have in 2024. This is our team research section, CCRF, CRU, and many people will be there in the future. Thank you and welcome to this section again.
Thank you, sir, for giving that 360-degree report. I request our director, Professor M. Srinivas, to address the gathering. Sir.
Thank you, respected dignitaries on the dais, off the dais, the young innovators and everybody. It's always a pleasure, see I think the research day is more important for all of us than the institute day. If you look into that, the kind of dynamism, the kind of, the Saraswati floating there everywhere, the posters and the people. Yesterday night, 12 o'clock, I saw people roaming here. I was just going out of the office, and then so many of the students, they were discussing about the posters and everything. This is something which is very, very encouraging for all of us. And I think let us together commit more to the thing, that with respect to the capacity building, with respect to the time, with respect to the grants, money and everything, and collaboration, what sir was saying. That is something which is very important, because I think we need to collaborate with ourselves first, within the units and the departments. That is something which is, we need to focus on that, And beyond the boundaries, again, unless we collaborate with the people outside, we will not be having high impact this thing. And already we are we at the ministry and at the ICMR level, we are speaking to the people that how they will designate that these are the 10, 20 areas of interest to the country. They will tell these are the the thrust areas where we need to do research. We will choose the topics and we'll choose our people or something like that, and then we will go ahead with that. And in addition to that, we're also trying for the PMRF scheme for our own students. That means, that we have gone through the SFC, and then I think we'll go beyond that. That will give us a little bit of more, not only the finances, but also the research grants and everything. Because at the moment, what is happening is that we have a system where we are choosing the students. I think it should be the reverse. The best of the students should come to us and then choose us. That what is that they want to do, they should come with ideas. That's something which is very important. At the moment, we say that, you come, join my lab, this is my work, I already published 10, 15, you are the 16th of this, 18th of that, continue with the same thing and all that. That's not right. That's not good for the institute, and that's not good for the country. I think we need the youngsters, the good people, the most meritorious people of the entire country to come and say that, well, this is the guarantee, that the salary is guaranteed, the fellowship is guaranteed, the research grant is guaranteed, the lab is guaranteed. They will choose me, whatever they have interest, they will choose the faculty over here, and then continue to do that. Then only we will be able to have a high impact research in us. I think we need to change a little bit of things. A lot of transparency has come in the selection process. We continue to do that. The merit should not be compromised, and then we will have, I think the clinicians also need to do more. I've been discussing with the sir that how exclusively some sort of research grants will be given to the people who are in the clinical sciences, because what is happening, there's no end for the patients, Whatever the number of beds we increase, whatever the OPD timings we increase, there will be always a patient who is waiting. I think we need to cut somewhere the line and say that, well, this much of percentage of this thing is for the patient care, this much is a percentage protected or limited or restricted or reserved, whatever you want to call. That's the time for the research. I think we need to do that, because at the end of the day, the Trinity mission of the institution has to be upheld, because when we go to the people in the ministry and all, they say that aapka research output kya hai? It's not the numbers, it's the quality and the impact which we give it to the public and the community and to the country, Though we are into the policy making of the country, and then we are into the major projects of the country, I think we need to enhance over there so that our presence is felt, and we continue to do a high-end research, so that people should not say that, we haven't got a Nobel Prize. I think that's where the youngsters has to see. We must get a paradigm shift over here, so that more time, more money, the more grant, more people, more collaborations will happen, so that we are in the reckoning, and then we continue to be in the number one, not only in the country, but we should enhance our ratings in the internationally also. Thank you once again for having come over here. Very thoughtful introspection for all of us. And then being an insider, what are the problems which we have and what are that which we need to do. And that's something which opens up our eyes for that. And then I think we must give a big hand to the youngsters, and we need to spend more time with them. See, the best thing, as a student, as a senior resident, I was always looking at somebody has to come and listen to me. I think that we must do the justice for that. Maybe we continued for more than whatever the days. Dr. Vinit is there. Up, samne baitho. So just spend time with them, bring more people, just spend with them, discuss with them, what is that they want, they will give some ideas, they will say what are the difficulties they are facing, what are the dreams and aspirations and the projects they have in mind, who is going to help them. I think we need to spend time, because there's no end, my patient will always be waiting. I think we need to spend more time with these people also. Thank you very much again.
Thank you, sir. without further ado, we will get on to the AIIMS Research Excellence Awards. The evaluation year was 2022, that is Jan 2022 to December 22. And there are multiple categories of the award. So based on the publications that were submitted by the faculty to the research section, there were AIIMS Excellence Awards in clinical sciences and basic sciences. Then there is an Infosys Oncology Best Paper Awards, again in clinical oncology and basic oncology. There's a new award this year, Dr. Surendra K. Chawla Award for the Best Paper in CTVS. And there are 2 seats or chairs that have been instituted as endowments. One is the Shobha Seth Wenger Distinguished Chair in Gynecology, Gynecologic Oncology, and the other is the Infosys Chair in Oncology. So there are a lot of prizes, and there is money associated with it too, which is not very important when we are getting adequate funding from different research funding agencies. So the number of papers that were screened for the Clinical Excellence Awards were 54. For basic sciences, it was 41 papers that were screened. For oncology, 21 papers were screened in the category of clinical studies, as well as for basic sciences in oncology, it was 14 papers that were screened. So starting with the commendations in clinical sciences, the first commendation goes to Dr. Sanjeev Kumar Gupta for his paper published in Hemisphere in 2022, Clinical and Prognostic Impact of Copy Number. So please, I request dignitaries to come on the stage to distribute the awards. Dr. Sanjeev Kumar Gupta. The next commendation goes to Dr. Ayush Agarwal for his publication in Scientific Reports in 2022, Primary CNS Vasculitis, a Cohort Study. Dr. Ayush Agarwal. The next commendation is to Dr. Sandeep Agarwal for his publication in Surgical Endoscopy, Impact of Bariatric Surgery on Urinary Incontinence in Morbidly Obese Individuals. The next commendation goes to Dr. Kritika Rangarajan for her publication in Scientific Reports, Ultra-High Resolution Multi-Scale Context-Aware Approach for Detection of Small Cancers on Mammography. Dr. Kritika Rangarajan. There is another commendation in clinical sciences to Dr. Avadh Kishore Pandit for his publication in Microorganisms, Mucormycosis in COVID-19 Patients, a case control study. The third prize in clinical sciences goes to Dr. Piyush Ranjan for his publication in the European Journal of Clinical Nutrition, Effect of Intensive Weight Loss Intervention on Metabolic, Ultrasound, and Anthropometric Parameters Among Patients with Obesity and Non-Alcoholic Fatty Liver Disease, a randomized controlled trial. There is another third prize, also in medicine, to Dr. Animesh Ray for his publication in Medical Virology, a study on the morbid histopathological changes in COVID-19 patients with or without comorbidities using minimally invasive tissue sampling. The third prize goes to Dr. Bishwaroop Chakrabarti for his publication in the Journal of Sleep Research, the Childhood and Adolescent Sleep Evaluation Questionnaire, development and validation of an ICSD-3 based screening instrument, a community and hospital-based study. The third prize also goes to Dr. Archana Singh for her publication in the British Journal of Nutrition, standardization and application of a novel multiplex assay for estimating micronutrient status and inflammatory markers in women of Sauria, Pahadia, and Santal tribes of Jharkhand. Another third prize to Dr. Deepali Jain for her publication in the Company of Biologists, molecular signature of postmortem lung tissue from COVID-19 patients suggests distinct trajectories driving mortality. The third prize also goes to Dr. Yatanpal Singh Balhara from the Department of Psychiatry for his publication in the Asian Journal of Psychiatry, a randomized controlled trial to assess effectiveness of GAM-E and e-health intervention to self-manage gaming with an aim to prevent gaming disorder. It's been received by Dr. Siddharth Sarkar, who is also co-author. The second prize goes to Dr. Sameer Bakshi for his publication in Lancet Oncology, Sex Disparity in Childhood Cancer in India, a multi-center individual patient data analysis. The second prize also goes to Dr. Kanaram Jat for his publication in Chest, Azithromycin for poorly controlled asthma in children, a randomized controlled trial. The first prize goes to Dr. Purva Mathur for her publication in Lancet Global Health, Healthcare-Associated Bloodstream and Urinary Tract Infections in a Network of Hospitals in India, a multi-center, hospital-based, prospective surveillance study. Dr. Purva Mathur.
We move on now to the basic science prizes. The commendation goes to Dr. Siddharth Kundu from the Department of Biochemistry for his manuscript, Temporal GSSA, a numerically robust R wrapper to facilitate computation of a metabolite-specific and simulation time-dependent trajectory from stochastic simulation algorithm-generated data sets. It was published in the Journal of Bioinformatics and Computational Biology. Another commendation goes to Dr. Uma Sharma for her publication in Clinica Chimica Acta for NMR-based metabolic profiling of patients with potential celiac disease, elucidating early biochemical changes of gluten sensitivity, a pilot study. The next commendation goes to Dr. K.H. Rita for her publication in neurological research, titled Dihydromyricetin Alleviates Cerebral Ischemia Reperfusion Injury by Attenuating Apoptosis and Astrogliosis in Peri-infarct Cortex. The next commendation goes to Dr. Ashish Suri for his publication in World Neurosurgery on the effect of the segmentation threshold on computed tomographic-based reconstruction of skull bones with reference optical three-dimensional scanning. Another commendation The next commendation goes to Dr. Pranay Tanwar for his publication in Computer Methods and Programs in Biomedicine titled Computational Investigation Reveals that the mutant strains of SARS-CoV-2 have differential structural and binding properties. The third prize goes to Dr. Manoj Kumar Tembre for his article Interleukin 33 induces neutrophil extracellular trap formation and macrophage necroptosis via enhancing oxidative stress and secretion of pro-atherogenic factors in advanced atherosclerosis. This was published in Antioxidants. We have another third prize for Dr. Dinu Chandran for his article, Differential Role of Central and Peripheral Arterial Stiffness in Determining Brachial Artery Resting Retrograde Flow in Patients with Ischemic Heart Disease versus Healthy Subjects. This was published in the Journal of Human Hypertension. The next third prize goes to Dr. Sujata Mohanty for her paper promoting in vivo bone regeneration using facile-engineered load-bearing 3D bioactive scaffold. This was published in Biomedical Materials. There is also a third prize for Dr. Devraj Singh for his publication in International Journal of Biological Macromolecules for his publication application of elastin-like polypeptide containing extracellular matrix binding ligands in regenerative medicine. A third prize also goes to Dr. Sachin Kumar for his article, Evaluation of the Programmed Death Ligand 1 mRNA Expression and Immunopositivity and their Correlation with Survival Outcomes in Indian Lung Cancer Patients. This was published in Human Cell. The second prize goes to Dr. Rupesh Srivastav for his publication, Bifidobacterium longum ameliorates ovariectomy-induced bone loss via enhancing anti-osteoclastogenic and immunomodulatory potential of regulatory B cells, published in the Frontiers in Immunology. Another second prize goes to Dr. Babaswata Nayak for his publication, Pattern Recognition Receptor CD14 Gene Polymorphisms in Alcohol Use Disorder Patients and its Influence on Liver Disease Susceptibility, also published in Frontiers in Immunology. And the first prize in the Basic Sciences category goes to Dr. Archana Singh for her research article, Chronic Hyperglycemia Drives Alterations in Macrophage Effector Function in Pulmonary Tuberculosis, published in the European Journal of Immunology.
So now we have the Infosys Award in Clinical Oncology, and this was a chair that was endowed by a grant that was given by the Infosys Foundation. And for this, in clinical oncology, the third prize goes to Dr. Anita Chopra for her publication, Hematology Oncology, Prognostic Utility of Key Copy Number Alterations in T-cell Acute Lymphoblastic Leukemia. The second prize goes to Dr. Sameer Bakshi for his publication in Clinical Nutrition, Prevalence of Micronutrient Deficiency and its Impact on the Outcome of Childhood Cancer, a prospective cohort study. And the first prize goes to Dr. Chandrasekhar Bal for his publication, Journal of Nuclear Medicine, Survival Outcomes in Metastatic Gastroenteropancreatic Neuroendocrine Tumor Patients Receiving Concomitant 225-AC dotatate, Targeted Alpha Therapy, and capecitabine.
The third prize in the Basic Oncology section goes to Dr. Jyotirmoy Banerjee for his paper, Transcriptomic Profiling of Non-Neoplastic Cortical Tissues Reveals Epileptogenic Mechanisms in Dysembryoplastic Neuroepithelial Tumors, published in Functional and Integrative Genomics. The second prize goes to Dr. Vaishali Suri for her paper, DNA Methylation Profiling of Meningiomas Highlights Clinically Distinct Molecular Subgroups, published in the Journal of Neuro-Oncology. And the first prize goes to Dr. Kunzang Chostol for a paper in Frontiers Immunology titled Upregulation of Atypical Cadherin FAT1 Promotes an Immunosuppressive Tumor Microenvironment via TGF-beta.
So this year there's a new award, Dr. Surendra K. Chawla Award for the best paper in the field of cardiothoracic surgery. And this goes to Dr. Suruchi Hasija for her publication in Journal of Cardiothoracic Vascular Anesthesia, Bivalirudin Anticoagulation in Neonates and Infants Undergoing Cardiac Surgery. A little background about this award. Dr. Surendra Chawla is a 1963 graduate of AIIMS who excelled in the field of CTVS. And after a long career in the United States, he was awarded the Lifetime Achievement Award at St. Francis Hospital. He retired from active clinical practice in 2018 and has continued to his research work. So this prize has been instituted by him, and I think his family members are also with us today in the auditorium. The next award is the Shobha Seth Wenger's Distinguished Chair in Gynecologic Oncology. Our previous recipient for this award was Dr. Neja Butler. She's here with us. I request her to come on stage to award to join the dignitaries on the dais to award the next awardee. And this year, the award goes to Professor Sandeep Mathur in the Department of Pathology. There is another chair that was established by a grant by the Infosys Foundation. And this year, the chair in oncology goes to Dr. Asima Mukhopadhyay, who is a consultant in gynecological oncology and lead in cytoreductive surgery at James Cook University Hospital in the United Kingdom. So that's the end of the awards. But now we have the release of the e-directory of the research section. And I request Professor Rakesh Lodha to come on the dais with his team for the release of the e-directory with the members of the editorial board, his team.
So ये on है, so you have to click it.
चलो अभी ताली बजाओ.
So please click it again.
नहीं चल.
So that's the link for the e-directory, and people who have submitted their profiles can now visit it. It's live. Those are some of the glimpses from the directory and some of the profiles. So thank you everyone for contributing, and thanks to the editorial team for making that great effort. Thanks a lot.
बीच में एक फोटो ले लो साहब.
Sir, would you like to say